continue to understand the R and how to manipulate and interpret your data. As I am seeing the numbers are increasing, I want to hand over to our facilitators to start the training. Professor Susan, over to you.
Good morning. Can you hear me? Good morning, participants. Can you hear me? Good morning, participants.
Yes, we can hear you.
Okay. Yesterday I left a simple assignment. Is there anybody who has done the assignment? You can just put up your hand and then you'll be given an opportunity to present. Okay. Hello, participants. I left a simple assignment and it would be nice to, if somebody did the assignment, please.
Wycliffe is already on, even in need.
Okay, we can have like 2 to present. Thank you. Go ahead, Wycliffe.
Thank you so much for the assignment we were given yesterday. It was about plotting 2 quantitative variables, 2 qualitative variables, and then a quantitative and a qualitative variable. So the first thing was to load the libraries necessary for running the exercise, which I believed tidyverse will be sufficient, and then another library called Patchwork, which I used to combine the plots. So line 2 and 3, if I run the 2, they load the 2 libraries. And then line 6 is going to help me to read the data. But I had the data in my project folder called data. So I just organized them that way. So I could see the data, the output and all that. So employee read.csv, then read_csv, then I have my data. I knew that I would use gender as one of the qualitative variables and also job category as qualitative variables. So I converted them to factors we were told in line 8 and 9. And then to plot 2 qualitative variables, I used ggplot for most of the plots, or for all of the 3 plots. So I supplied the data into the ggplot function, and also aesthetics. I provided one of my categorical variables, and both quantitative variables, salary and salbegin. So, and also the I used the geometry which I used to map this data on the plotter is geom point. I also attempted to smooth it and then added labels and font size of the text element to some legible one. So if we run that, we had this kind of plot output. which I call which shows both quantitative variables, quantitative in the y-axis, quantitative in the x-axis. And we could fit that ggplot object to P1, to a variable name P1 to refer to my first plot. Secondly, two qualitative variables. So in this case, one would be plotted and then the other one would be used to fill it by some color. So again, I use ggplot. I supplied the data employee and then aesthetics of job category filled by gender. So we only have x-axis in this case. So the y-axis would be the counts in each category. Then I used geombar and not geompoint because they are not continuous variables. Labeling and setting them to element size to 12 remains almost the same. So in this case, we could see that in clerical job category, we have almost equal number of representation of both gender, but in custodial, we only have male, and in managerial jobs, more of male and quite few female. So both are qualitative data. On the y-axis, these are just counts of number of male and female in each job category. So lastly, quantitative and qualitative variable. So again, I supplied data of employee to ggplot function and used aesthetics. In this case, I went some step ahead to reorder my gender by logarithm of some salary begin variable. So this can be a bit strange for those who have not used it before, but it's just ensuring that I order my plots to look pretty fine, and also log transforming the cell begin to bring down the values to some smaller values on my x and y axis. The geom I used here is box plot because it could bring the continuous variables under each categorical level pretty well. So this is what I ended up with: beginning salary versus gender of the professional. Then I could supply that to P3 as the third plot. Here then came the work of patchwork. And patchwork is just to organize all the plots together. So P1 will be above P2 and P3, as simple as such. Then we can annotate the patchwork using some tag levels to show the labels. Like if this were figure 1, then it would be figure 1A, the other one would be figure 1B and C. So I have my line plot of both quantitative variables on top, and then qualitative and qualitative below, and then qualitative and quantitative also below my plot. You can reorganize them. All of them can be on the same row, all of them can be on the same column, and all that. Then ggsave function is just to write this output into my folder. But then because it's an output of my analysis, I also had an output folder where I throw the plot into and set DPI to 300 to make it better legible to somebody who will be interested in reading it then. So if I go to my output data, you can see I have my 3 plots. And if I open it, I can see all the 3 of them. Yeah, so that's all I did yesterday.
Thank you very much, Wycliffe. That was excellent. I think participants have learned new things that we didn't even mention, like patch. And I hope you will be able to use that knowledge in putting the graphs together and presenting them in a document, and also saving them onto your folders. Thank you very much, Wycliffe. Please give a clap to Wycliffe. Let's have another person share. Yes. Niche. Okay, Mitch. Neat. Okay, I'm told Neat is the second person who will be presenting. Please, you can start sharing. Neat, start sharing. Can we have Neat share the work?
Neat, please unmute yourself. Okay, so we go to Mahmoud, since Neat is failing. Mahmoud, go ahead.
And share.
If Mahmoud is failing, we have Ayana Olana, or Mona Mohammed. Any of them can go ahead.
Thank you.
Ayana Olana or Mona Mohammed. Unmute yourself and you share the screen. Yes, Ayana, you have switched on the camera, but the microphone is still muted. So unmute yourself and go ahead. Ayana, Olana, please unmute yourself. Go ahead. You are now unmuted. Okay. I think he has dropped off. So Mona, you can go ahead. Ayana dropped off, so you can. Yes. Okay. Mona is now on.
Okay. Thank you.
Good
David.
morning, bro.
Morning, Mona. Go ahead and present.
Yeah. Okay. I'm sorry, I'm beginner. I'm not professional. Like my first colleague. So I tried, just tried. I installed the packages. Then I load it. Then I read the employee as CSV. After that, I run to know the relationship between variables. I run this command to see the relationship between the salary and job time as two qualitative variables. After that, I run to see the gender and education as two qualitative variables, and after that, run this command to see the relationship between gender and salary, quantitative and quantitative. This is all what I done, Professor.
Okay,
Thank you.
thank you. Thank you, Mona, for a good try.
Is it correct, Prof?
For the gender and salary, the best summary statistics would have been a box plot or a bar graph, but or a bar graph, a box plot or a bar
for
graph
gender and
for gender and salary. Okay. Then salary and
gender
job
and
time.
salary.
Gender and salary is a box plot.
I did this, Prof. I did
Okay.
this one.
You can run that one. Yes.
This one.
Yes. Okay.
This is
yes.
better.
Yeah, that is better.
Okay. Okay.
And
Thank
salary
you,
and
Prof.
job category is also box plot.
Okay.
Okay.
Thank you.
You're welcome. But thank you for a good try. That is very good.
Thank you. I'm beginner, really. This is first time. This is first course in art for me.
Oh,
thank you for giving me this opportunity. Thank you.
You're welcome, Mona. Thank you for being brave and presenting what you've done. You will learn. You will improve. Okay. I think we give clap to Mona and Wycliffe for the presentation. I hope we've all picked that when you have a quantitative variable, two quantitative variables, what you want to know is the relationship between those two variables because they are both continuous. And if you want to look at two qualitative variables, then we can either do a cross-tabulation and draw a bar graph, okay, to summarize and present two qualitative variables. Then when it comes to one qualitative variable and a quantitative variable, then we have to use a box plot or a bar graph. If you want to use a bar graph, then you have to summarize your data by the categorical variable and use the mean to construct the bar graph, not the frequency. So remember that the type of variables I'd mentioned determines the type of summary statistics or descriptive stat you're going to use. So we had one more command to run. I want to share my R script and you also got your R. So once we finish that small bit, then my colleague, Dr. Thomas, will come in after that. So I'm going to share the R script. There we are. Okay, so I wanted it to become a bit visible. And the last part which we want to share, the first thing I will do is to load the packages. As usual, we have to load the packages. So I'll go here and load the packages. Okay. So you do the same, make sure you load the packages. Then the second bit I'm going to do is to set directorate. So I'll go to session window, set working directorate, choose working directorate. So then I go to the folder where I'm working and it's day 2 and I'll click OK. So my directorate has been set. Then the next thing I do is to bring in the data set. So I'll call employee data set using line 30 and 31. So I run those lines, and head means you want to view the first 6 data points of your data. So from there, then we go to the last bit, which we didn't run yesterday. Those were the plots, I think. We are done correlation. So we wanted to construct multiple graphs, and it is line 225, where you give an option so that the window is arranged in such a way that you want 4 graphs per row and column. So this is the command which I am first putting, which is pow, then mfro, combine C, 4. So I'll run that and then run the plot. It says object employee not found. Okay, I had to subset, sorry about that. I need to subset the data first, because remember that employee 1 is the data set which has only continuous information, and it's the one where I want to run multiple graphs. So I go back to line 195 where I subset my information. So I run line 195, and that one removes all the categorical data and we remain only with continuous. Then I'll go down to that graph part. So I run the plot part, and there we go. So I'm going to zoom in so that we see our graphs and share. So let me share the graphs. So those are the graphs. I wanted to draw multiple graphs, and the option was just to prepare the window to take 4 graphs on the rows and the columns. You can see that it is telling us that this first column is for education. So we see that education, and the second row is salary. The third one is salary beginning. Then we have job time and previous experience. So if you want to see the relationship between education, number of years you've spent in school, and current salary, this is the plot here, which shows a linear relationship, but not very strong. Then if you want to find out number of years spent in school and beginning salary, the graph is the same here, graph number 3. And it also shows a linear relationship, which is not very strong. Then when it comes to education and job time, that is the time you spend in school, it is just a random scatter of points, meaning that there's no linear relationship between education and job time. So we don't expect to find that those who have spent more years in school are spending more time on the job. There's no really a relationship. And then the last one is previous experience and education. Also, it's just a random scatter of points, no relationship. So we can also look at column number 2, which is current salary. And you can see this same plot is the same one you had here, but it has been flipped in such a way that salary is on the x-axis while education is on the y-axis. That's why you see it has been changed like this, but still showing a linear relationship and a weak one. Then when we come to salary, beginning salary and current salary, we've already seen that it has a very strong linear relationship between current salary and beginning salary. When we come to current salary and job time, the relationship is there but weak. It is a positive relationship but weak. When we come to previous education and current salary, you can see that this relationship is not positive, it is negative, but it is also a weak relationship because most of the information is just clustered in one section. Okay, so you can continue like that, looking at different variables, but you've drawn them in the same window. And if you want to save this, you can take it to your Word document. So let's just see, stop sharing that. And I share the script. So if you want to export this window, the plot to your document, you use this export command here. Under the plot window, there is a word export. So if you click on export, you will see that it has save as an image, save as a PDF, or save to clipboard. So whatever you use can save. So if we click save to clipboard, you can see it is here, and then you can copy the graph. So here we copy the graph and then open the Word document. I'll paste it, then I'll show you that she's already there. Control V. So then I've pasted it, so I'm going to stop sharing and share what I have pasted in the Word document. So this is what we have in the Word document, and you can now write around figure 2 or figure 1, a plot of many things, and then many variables, and then write around in your document. So if you want to take it as a PDF, then you export it as a PDF. So hopefully that is clear, exporting the graph. You go under, I'm going to share again and repeat it. The script, you go under the plot window, there is a word export, and when you click the arrow pointing down, it gives you 3 options: save as an image, save as a PDF, and save to clipboard. So if you save an image, it's going to go to your folder, and then you can insert it later in your documents. Okay. So I think we've already done cross-tabulation, but we can repeat it. The last part is cross-tabulation, and cross-tabulation is for categorical variables. So we want to cross-tabulate gender and job cut. So we've named that command SA. The object is named as SA, and we are using the table command to do our cross-tabulation. So if we run that and run, you will see that you have a cross-tabulation in your console of the job category and gender. So if you want to convert those frequencies to percentages, then you can convert them to proportion using the prop.table command and then round off to 2 decimal places. So we run that, and it gives us the table with the percentages rounded to 2 decimal places. So you can now copy this table, take it to your Word document, and write around. The highest number of employees are cleric female and also cleric male. The least number is a female manager. You can see it's 2.1%. Okay, and there are more male managers where we have 15.6% compared to 2.1%. Okay, so the rest of the commands is for you to clear if you want to clear your console. Then you run that, and Control L, you do it on the keyboard. So the rest you can actually do them at your own pace. I want us to benefit from what Dr. Thomas is going to present. I thank you very much for giving me this opportunity to share with you this morning. I give the mic. I'm going to give it to Dr. Thomas to take you through generating experimental designs and surveys. Thank you very much. Unless if there's a question, let me check in the chat. But I believe there are no questions I can see. Thank you, thank you. And let me hand over to Dr. Thomas. Thank you very much. Okay, bye-bye.
Can you stop? Hello, good morning, everybody. I hope you are enjoying yourself, and we'll continue with the process. So I'm going to take over and just have a bit of a theoretical introduction to design of experiment, and then we run some few scripts. Those scripts you can as well even run it yourself. So I need to share my screen.
My speaker, my speaker is not working.
Just hold on a little bit. Thomas, could you be talking and I will just mute it, or because we?
No, sorry. No. I muted myself because I was sorting out some issue, but I'm back now. Hello? Okay. So I want us to look at the design of the experiment. So before that, we can have just a brief look at the research process. So we all know what research is, but I just want to reinforce a few things. The first thing is that research is a systematic process inquiry. So that means there's supposed to be order. Somebody is supposed to do to repeat your studies without any problem. Entails the collection of the data, documentation of critical information, analysis and interpretation of the data of the information. And then there is supposed to be issue that the methodologies that you use are supposed to be suitable. And then the design and everything else will depend on your professional fields or your academic discipline. So you'll find there are differences between between different groups. So if you are in social science, you're probably going to have slightly different methodology to compare somebody in fisheries or somebody in agriculture and so on and so forth. But generally the principles remain the same. Research also is kind of a cyclic process. So it takes on a cycle. It's not something that is going to move in a straight line. At some point you move and then you come back. So we always start with say you start with problem identification. After identifying the problem, we all know, those who are studying, that in your thesis, you are expected to have a statement of the problem. Once the problem is clearly identified and clearly stated, the next thing is you need to break up your problem into subunits or sub-problems. And this is where your research objectives will be derived from. And then in relation to the research objective, we have research hypothesis or research questions. Based on the research hypothesis and research question, one is there for us to go to the field to do some data collection. After you've done your data collection, you do the analysis, you draw your conclusion, and from there, you never know, you might have identified new problems, and then you state the problem and the process continues until on and after somebody will pick up from where you've actually done. Okay, so what we do, the processes that we study as scientists, whether you're a biologist, you're a social scientist, or you're an engineer, a natural scientist, we are dealing with a lot of variation. We study variation in order to understand relationship between different factors. So the variation that we observe in our data mainly come in two forms. There's variation that you as the experimenter or as an investigator, you evoke that variation by what you do. You manipulate the situation. So if you are an animal scientist, for example, you can use different breed of animals. So using different breed of animal means they will respond differently to different situation or different conditions. If you're a plant scientist, you may have different varieties. If you're a soil scientist, you may want to use different fertilizers. If you're a chemical engineer, you may use different chemicals. If you're a food scientist, you may use different food preservatives and so on and so forth. So whatever when you change the condition of a system, you expect that the different units that you're studying, you expect them to, just hold on a little bit. Hello, I'm back. Sorry, I think there's just some office issue that we're finally sorted out, so that will be fine. As we study any process, any systems, I indicated that we always generate variation. The other variation is always not under your control as an investigator. So there are things that you can change, but there are things that you cannot change. It's difficult for you to control altitude. Of course, you can go and do your study in a certain altitude, there are things like temperature, there are things like soil textures. So these are things that you may not be able to control, but in one way or the other, you see how to organize your study or design your study so that the effect of these different factors are taken care of, so that whatever you're going to study, you're going to study without having a lot of what we call confounding that I'm going to talk about later. Of course, as we do research, there are processes that we go through. So the first thing that we always do is that you do what we call a problem identification. So you need definitely to come clearly and identify the kind of problem that you're interested in studying. So the problem, it could be kind of a scientific interest. So we do what called basic research. So people have been doing research on how to get to the moon, to go and sample rocks on the moon. Of course, this sampling rocks on the moon are not going to solve any immediate problem. It's not going to end hunger in Africa next year, but it might at the end of the day contribute to greater improvement in the condition of human beings with time. But most of the research that we do are practical concern. That's what we call applied or adaptive research. So you need to identify which of this area you are interested in. So after you've identified your problem, the next thing is you are supposed to do some literature review. So with the review of literature, what you want to know is you want to know in relation to this problem that I'm interested in studying, what is it that is known? What are people done? Even in statistically, you want to know what are the methodology, what method do people use for analyzing such kind of data. So you need to know exactly what's known about the subject you're trying to study. And then the next thing also, you want to know what is not known. That is the gap, because you want to study something that has already not been studied. Otherwise, then that will be a duplication. No one is likely to have to give you a degree if you're duplicating some work. The next thing is then you define your objective or study objectives. And this is what we say now. We look at the problem that you've identified, and then we break that problem up into smaller parts or sub parts, and then we define our objectives based on the different sub parts of the problem. So as you study, research objective is one of the most important stages of your research, because everything else that you're going to do after that will depend on what you define as your objectives. So you need to come up with a specific objective in relation to your research problem. After you've defined your objectives, and in some cases, actually, the hypothesis comes first, and then the objective comes out of the hypothesis. So you need also to define your hypothesis very clearly. So hypotheses are just theoretical propositions, or what I would say, a tentative guess to the answer of the research question that you're trying to address. So it's an educated guess. Your guess will depend on your experience. Your guess will depend on how much you know about the subject. Your guess will depend on how much you've read from the literature. So hypothesis is very important. We're going to spend a bit of time looking at what hypothesis is. It's important for the kind of data that you collect and the kind of factors that you're likely to include in your study. After you've stated your hypothesis, of course, hypotheses need to be tested. So the next thing that you need to do is to go and collect data from from the field. Now, so this is where the main topic of today is going to come from. So we need data that will provide evidence for or against the hypothesis that we have already set. So you can either generate your own data by means of experiments or research, or you can also collect data, get data that is already in use. I mean, people have collected data. You can go, for example, to the National Bureau of Statistics of your country. You find data that has been collected, what you call secondary data. You can also use that data there to actually test your hypothesis or generate new hypotheses. So the design of the study, whether survey or experiment, will certainly determine the results of the quality of your results. So we need to design a suitable study in order to generate data that can test our hypothesis. If you don't generate the correct data, there is no way you're going to generate science. So your result is as good as your data. If you have bad results, the data, I mean, if you have wrong data, you're going to end up with completely things that will not make sense. After you've collected your data, of course, we looked at the analysis of the data. We already started doing exploratory data analysis. My colleagues have already shown us how to do exploratory data analysis. And then we're going to continue how to do what we call inferential data analysis and so on. Of course, after you've done your data analysis, one would be expected to draw conclusions, to interpret the results. So interpreting the results would certainly depends on your knowledge of statistics. It depends on what you've read in literature. It depends on your expertise as a person in the field. So under interpretation, we would expect you to draw logical conclusions. You need to make inferences. You need to tell us whether you have developed new principles or new methodologies. You need to tell us whether you found a solution or not. And then as you write your thesis, sometimes you also need to acknowledge the limitation of your results. I mean, we would want to achieve certain things, but based on what we have in the field, we may actually fail to achieve that. Then of course, you do dissemination. If you're a student, you need to write a thesis, you need to publish. If you're a staff, they say, publish or perish. So if you want to get promoted, you need to publish your result. So just for emphasis, I want to just talk briefly about around the roles of objectives in your study. There is no way you're going to progress with your research without having defined clearly the type of objectives that you would want to achieve. So with a clearly defined objective, the researcher can focus on the study, because then you know the direction which investigation is going to take. Sometimes if you go to collect your data in the field, if you don't have your objective, but you may end up actually collecting the wrong data. Sometimes what happens is we pick questionnaires. Let's say I picked questionnaires that Ellen has used, and then because I say, well, Ellen is a very smart woman, so given that she has already done this, I can pick that and use. So maybe she's doing a study that requires her to know how many goats or how many dogs people have in their villages. I pick that and I don't change it. I go to, then I start asking people how many goats they have, and yet it may actually have no relationship with what I'm studying. So what we need is that whatever type of data that you're going to likely be related to the objective. So objective should be clearly stated in a way that will enable you to collect your data, collect the right amount of data, the right type of data, and at the right time. It also helps you to organize the study in a clearly defined parts of phases. For example, you can say, well, objective one would be to look at the variability that exists maybe among I mean, if let's say you are a plant pathologist, I mean, you're interested in plant diseases. So you may want to know what are the different types of organisms that causes a particular disease. Now that would be your first step. After doing the first step, then getting the isolating maybe those pathogens, you can then do something with them. So objective will help you to organize your study in a clearly defined parts of phases. And it's always expected that your objective should actually be linking up, should be related to each other. If one objective is talking about determining the effect of eating food on the height of children, the second objective should also be related to that. If you're going to bring something that's completely out, that already shows that there's some kind of disorganization in your study group. Of course, it also helps to facilitate development of research methodology. So in a way, from your objective, you need to ask yourself, do I need an experiment? Do I need a survey? If I need a survey, I'm going to design a questionnaire. What kind of question do I need to ask my respondents? If I'm interested in determining the effect of poverty on say, the number of children that somebody a family will have, then I need to know, how do I measure poverty? Okay, how do I determine the number of children in a family? In some cultures, you don't count children. So if you come to a man, a head of a household, and you ask them how many children you have, they are not going to tell you. They say, no, that is a taboo. So how do I get around those? So maybe you can say, well, you go there and you say you're distributing sweets and the kids will come around, and then you'll be able to get that. So it facilitates development of research methodology, helps to orient data collection, analysis, and interpretation of your data. So the objectives of your study are very, very critical for the success of a given study. Of course, any experiment that we do, people would expect us to have what we call research hypotheses. Research hypotheses are simply kind of answers to the research question that we posed. So hypotheses are developed by asking the what, why, how, when, where, and what if of the different aspects of the causes of the problem. And once you ask those questions, if you're going to produce a tentative answers to those questions, those ones are stated in terms of hypothesis. And as I indicated with the research objectives, hypotheses are equally very, very important as far as guiding your studies concerned. So a clearly stated hypothesis will help to head and determine the design of the experiment or survey. So if your hypothesis is not clear, there is no way you're going to design a good study. It will help to identify what are the appropriate treatments and what are the control. So what are you trying to compare? So if your hypothesis is clearly stated, you should be able to actually tell what type of treatment are you going to involve in your study. The next thing is going to help you to determine the type of data that you collect, and then probably going to suggest the methodology. Now, this brings me to what we always do. As statistician, we love what we call the null hypothesis. So the null hypothesis always say there is no difference between A and B. Now, the null hypothesis is not very good at this stage. At the stage of your writing proposal, you need what we call researcher's hypothesis or the alternative hypothesis. That's the one that is certainly going to guide you carefully. If you're going to write a proposal to me to fund your study and you're telling me there's no difference between what you're going to study, I mean, why would I waste my money on that? So the null hypothesis, you can leave it for the analysis stage. That's when you have your null hypothesis. But when you're writing up your thesis or writing up your proposal, it's important that you state your hypothesis in a way that is going to give you the direction of the study clearly. I'm going to show you one example how to develop an hypothesis. So hypothesis is derived from a research problem, which is usually framed as a question. Okay, so I have a problem here. Let's say, for example, that farmers in your area or in your country, they're experiencing low yield of a particular crop. In this case, let's say this is groundnuts. So farmers are experiencing low yield, and now there's lack of information on how to improve the yield of this particular variety. So this is our problem. How can we generate or formulate hypothesis out of this so that the hypothesis that we generate is able to guide our study, like what we talk about as the importance of hypothesis before, okay? So possible question that can be asked here, why are farmers in Uganda getting low yield when they grow this variety? That is a research question, okay? Or another alternative, you could say, how can we increase a yield of Acholi white in the farmer's field? So you see there are two questions here. Now, your hypothesis can be formulated as an answer, a tentative or a guess to the answers to these questions. And you need to state it in a way that will actually push the problem, the solution to the problem forward. So how do we formulate hypothesis? How do we start? So in developing hypothesis, you need to know you are influenced by an existing theory. So we assume that you're not working in a vacuum. Maybe lots of studies have already been going on in this particular area that you have. So we assume that there is some existing theory. We assume that related research has been done. If you actually been working with a crop or animal or people for some time, we assume that you have some personal experience. So based on this, you can actually have a rough idea what the solution to the problem is or what is causing the problem. So let's say, for example, your idea as a researcher or as a groundnut breeder is that this variety has actually become susceptible to a disease. Okay, that is what you think is causing the problem. So this is already now part of the, remember here we're saying, why are farmers in Uganda getting low yield when they grow this particular variety? So you already have an idea that possibly this variety that was introduced maybe in Uganda in 1960s or 1970s is already probably having a problem in that it has become susceptible to this groundnut rosette disease. Possibly this disease was not there before, or it's also possible that this disease was there before, but the causing agent of this disease actually has evolved over time, so that they are now in position to attack the variety that once was resistant to them. So this is going to be idea on which you're going to build your hypothesis. Okay. So possible hypothesis here would be that resistant to groundnut rosette virus disease in Acholi white is broken down due to emergence of new strain of the virus. So, and you cannot have this type of hypothesis unless you have some existing theory, or you read in literature, or you have some personal experience with that. So that means hypothesis cannot come out of the blue. You shouldn't be able to say, okay, I'm going to talk about this hypothesis. How do I come, how will I really get enough information to make me formulate a decent hypothesis that can be tested? So here is a possible hypothesis. So now the next thing that we're going to ask ourselves, can we test this hypothesis? If we want to test this hypothesis, what type of data do we need? Okay, how do we design an experiment that will help us to test this hypothesis? How do we use statistics to analyze that results in order to actually confirm or reject this hypothesis? Okay, so we need to go back and look at our hypothesis here and ask ourselves a few questions. can we design a study or an experiment or survey based on this hypothesis? So can I ask, for example, what kind of study can one do based on this hypothesis here? Can you provide some answers in the chat? Some answers. I want to see some experimental. How do we do the experimental? So we say, well, we can do an experiment. Well, that's fine. We can do an experiment. What else can we do? You're having sound echo? Hello? Can you hear me? Okay, somebody say we look at the DNA, we can do comparative study. Okay. Okay, good. So can we identify the kind of treatment? If you're going to do an experiment, what kind of treatment do you think we are going to have in this case? What kind of treatment? Yeah, you put the answers in the chat. We compared crop variety, different varieties. Okay, we have some good ideas. What type of data will we collect? Can I see in the chat what kind of data you think we should collect? Disease, yield, DNA strain, disease severity, good. Growth rate, virus incident. Okay. Okay. So what we see from here, that at least you are able to say something about the design, about the treatment, about the type of data collected, and probably you're able to say something about the methodology that is used. So if your hypothesis is not stated clearly, there's certainly going to be a problem as far as that is concerned. So please ensure that your hypotheses are as simple as possible, clearly relate to the problem to be addressed. So if we are talking about yield, for example, then our hypothesis should also be related to yield losses. It should be stated in a way that suggests the solution. It should be stated in such a way that provides direction for the research. And then it should be stated in a way that we can verify or reject the hypothesis, and then it should be stated as simple as possible. So these are some simple rules that one will need when you're stating your hypothesis. So I'm going to request you that maybe as we move along, you try to come up with some research hypothesis that we can have some time and discuss maybe early in the morning before the new day begins. Okay, so we assume now that you have your research objectives, you have your research hypothesis. So the next thing that we are going to do now is you need then to design a study that you can use to test your hypothesis. So the first thing we're going to look at overview of experimental design. So this is going to take the greater part of the day, and hopefully you're going to gain from this. So I believe that most of us know what an experiment is, but experiment is the only sure way of establishing cause-effect relationship. If you want to find out the effect of fertilizer on yield of a crop, the easiest and the most sure way of doing that is to set up an experiment. So the principles of the design of the experiment are the same, irrespective of which field of study. So whether you're doing agriculture, you're doing biology, you're doing ecology, social sciences, economics, as long as you're doing an experiment, there are basic principle that one needs to follow. And we're going to look at these basic principles of experimental design. I want, by the end of the day, you should be able to design an experiment. Those who do agriculture know very well field experimentation. So here you can see there are crops that are grown, and then there are trees here. There's some kind of a whole amount here. So based on this experiment, what can you tell? I mean, just tell us anything that you see from this experiment. From this, what you are seeing here, what are what do you think is being compared here? The shaded side is healthier. Okay, we are probably comparing different genotype, shadow effect, irrigations, light effect on growth rate. Okay. Okay, what would be a possible hypothesis here now? Based on what you think we are studying, what would be an hypothesis? Effect of treatment on growth rate. Somebody said PAR. What is PAR? Effect of light on plant growth. Effect of tree. Okay. So that is now photographic interpretation as we have there. So we also have laboratory experiments. So when we are talking about experimental design, there are two basic components that we are always talking about. We are always talking about experimental material and then experimental treatments. So there's treatment and then there is material on which the treatment will be applied. Okay, so if we look here on the picture on my right here, we see there are different animals here. So if you are somebody who is in vet or in animal science, you're always familiar with an experiment involving animals. So the animals in this case will constitute our experimental material. So each animal here could be an experimental unit, so the variation between the different experimental material or experimental unit as we observe here need to be taken care of when you're designing an experiment. And that's why the design of an experiment has one key component related to the structure of the experimental material that you have. And here the interest is mainly in controlling the variation outside the control of researchers. So if I go back to where we started from, we talked about the two sources of variation. We took at the variation that is evoked by investigator, so which could be the animal breed, plant species, fertilizer, variety, chemicals, and so on and so forth. And then we talked about the other that is not under the control. So this is where the material, the condition of your experimental material will be taken care of here. So the design should be able to take care of the fact that experimental material are always not uniform, and you want to try as much as possible to control that. Just hold on a little bit. Okay. So this is our experimental material, and that is one component of the design that we need to look at. You need to ask yourself, I'm going to do my experiment using this particular material. What are the issues that will come out of that? Then the other component, the treatment. So the treatments are always the condition that you want to compare. So this depends entirely on what you're trying to study. So if you are In social science, for example, people study a lot about these computer games and different types of the effect that those computer games have on maybe the ability to retain things, or effect. You could also have things like effect of sleep on performance and all that. This will constitute what called treatment. So I as a young child, when we were growing up, when we talk about treatment, we always talk about the hospital. So the drugs here, for example, can be the treatment that we want to compare. And then we want to compare using these animals here. So we could use the drug and see how the animals react to the drug. The animal constitute our part of our experimental material. The drugs constitute our treatment. And then when we look at these two together, will constitute for us what we call the design of the component of the experimental design. I have this simple example that I used to illustrate the different terms that are used in the experiment. So the first thing is what we call experimental unit. So experimental unit is always defined as the smallest unit of the experimental material to which the treatment is applied. The treatment, we said, is the condition that you're studying, that you're trying to compare. So for example, you may be interested in comparing different types of fertilizer. Maybe you're interested in comparing organic fertilizer with an inorganic fertilizer. Maybe you're interested in comparing the different pesticides and how they will react. I mean, they say, for example, your home has been invaded by mosquitoes, and you want to see which insecticide on the market is the most effective. Then what you're comparing, the condition, the room that you're going to change using your pesticide, your insecticide would actually be the treatment. So the treatment here is what we are trying to compare. So here we assume that our experimental unit is actually this pot here with the soil in, and if we're going to apply fertilizer, this is where it's going to be. And then, of course, after you apply the fertilizer, the plants have grown, you want to do some measurement. Now the question is, am I going to measure everything on all the plants here, or I'm going to pick one or two plants? So each plant here can be what we call a sampling unit. So that if you want to make measurement on the effect of fertilizer on plant growth, you can sample one or two plants here. Remember, when you're doing the sampling, you're also causing variation. Because if you sample these two and somebody else sample the other two, there's going to be variation between them. So that is also going to cause some kind of variation and on the result that you observe. Of course, then once I decided that, okay, I've set up an experiment, I've grown my maize, now I want to take measurement. I want to answer the question, is fertilizer really affecting the growth of maize? So the next question then, what do I measure to actually make the comparison? So things like plant height, number of cobs, leaf length, number of leaves will now constitute my variables that I have. Now it's very, very important to understand or to differentiate these different components if you are to be successful in doing an experiment. Because if you're going to mess, you cannot differentiate between a variable and a sampling unit, or a variable and an experiment, then we are likely to have a very, very big problem. So the first thing that we need to know is, can I differentiate what an experimental unit is? Because defining an experimental unit is important, because it's going to determine the type of design that we're going to use to study our treatment, or to compare our treatments. Let me see if there are some questions. Oh, somebody's still talking about installing R. Okay, so the next thing is, then we say, okay, if I'm going to do an experiment, what should a good experiment do? Okay. I always apologize for these pictures before I start talking, because it might offend you, but I'm just trying to illustrate my point. And so let's say in this case that we are comparing two energy drinks. There's what we call the Monster, and then the other one is Red Bull. This is our treatment. Now, our experimental material are human beings. So these are our experimental material. So when we're talking about design, we're looking at our experimental material, we're looking at our treatment. Okay, those are the component of the experimental design that we are talking about. We want to compare the treatment. Okay, that is Monster versus Red Bull. Okay, so a good experiment should provide for us unbiased estimates of the difference between the two treatments. Okay, so what is bias? I can define bias as a systematic tendency of a sample to misrepresent the population. Okay, so I want to see between these two conditions, set experiment A and experiment B, which one would give me a bias comparison? Can I see in the chart? A will give us a bias comparison. Okay, everybody agree? Why would A give us a bias comparison? Why would A give us a bias comparison? Somebody said the child effects, the age effects. Okay. Experimental units are different. Okay. I think I'm preaching to the converted, I think. Okay. So from here, you can actually see that what we call confounding can come up here. So let's say for one reason or the other, this gentleman, this small gentleman there wins the fight. Okay. Of course, the question will then come: did the gentleman win? Did the small gentleman win the prize because the fight because he took Red Bull, or he won the fight because he can easily move around and get the other gentleman tired, and then we'll be able to put him down at the end? So can we differentiate the effect of the experimental material from the effect of the treatment that we are comparing? Can we do that? Okay, so what we see here, there's what we call confounding happening here. So the difference, the variation due to the difference in the experimental material cannot be differentiated from the variation caused by the differences between the two treatments. So in this case, this A is likely to give us a biased estimates of the difference between the two treatments. So the bias could be in favor of this gentleman here, or could be in favor of this gentleman here, depending on what is involved. Okay, so here, this is most likely going to give us a fairer comparison, at least based on what we observe visually. The other thing that also a good experiment should give us is what we call experimental error. So experimental error is defined as variation between similar experimental units receiving the same treatment. Variation between similar experimental units receiving the same treatment. So what we see in this case here is that each of these rats here is our experimental unit. I'm giving them the same drug, same dose and everything. But when I'm going to measure their responses, it's very unlikely that all of them are going to respond the same way. Okay, because the experimental units are assumed to be uniform, and they all got the same. So any variation that we observe here, we can consider this variation here to be just a natural variation that will happen within the system. So this is what we call white noise. In statistics, they call it white noise, or in okay. Now, why is this important? This is important because when we are comparing treatment, we also need to take into consideration the fact that there is some background information. So we should be able to estimate this background information so that when we compare the variation that we think are due to treatment with variation that we think are due to due to the background noise, that ratio there should give us an indication of how strong the variation due to the treatments is. Now, if you cannot estimate that variation, that means you cannot do any testing. Okay. So for us to get these two things, we need unbiased estimates of differences between treatment. We also need unbiased estimate of experimental error. So this is another scenario. So here I have my beautiful dogs. This time, instead of using these rats here, which are uniform, I use these dogs here, which appear not to be uniform. So the same experiment, same treatment, but here now, if we make assumption that the experimental units are uniform, therefore this variation here, I need to get a pen. Okay, so we have this variation here. Now this variation is slightly different from this variation here. Okay, at least we can see this variation is much smaller compared to this variation here. And when we are here, we say because the experimental units are uniform, because the treatments are the same, we can assume that this is just a mere background noise. But here, it's probably, it's not really, I'm not very comfortable to assume that these experimental units are uniform. So part of the variation that we observe here as a result of differences between the experimental material. So there is the background. Okay, so we have the background, but we also have, there is also material effect. So this means if we don't remove the effect of this material from our analysis, then this noise here is going to be highly inflated, and this noise here is not going to allow us to test the differences between the treatment well. Okay, so therefore we need to design our experiment in such a way that we are in position to provide unbiased estimates of the differences between the treatments, as well as we need also to estimate our experimental error without any problem. So these are the three principles of experimental design. You probably heard people talk about replication, people talking about blocking, and people talking about randomization. Okay, so we're going to look at each of these and in detail. And once you understand this properly, you should then be able to have no problem in designing a suitable experiment. So what is a replicate? Why do we need to replicate, and how do we do the replication? Okay. So for definition, replication is simply repetition of a treatment under identical condition in an experiment. So if we have treatment A should appear in our experiment more than once. And then each treatment is applied independently to 2 or more experimental units. Now this part of the definition is very, very important. Okay, so look here. Here, we are applying this treatment here to this rat, independent of this treatment, to that rat, independent of this, to that rat. So each of this is an independent unit. This is an independent unit. This is an independent unit. And this is an independent unit. So for us to talk about replication, we need that each treatment should apply it independently. When we come here, the gentlemen are all enjoying the thing together. So that's not independent of each other. So here, application of treatment is not independent. So this is what we call pseudo replication, or what we call subsampling. If we are coming to sample, that will be a different case. Just similar to my maize example here. See, I cannot start saying this is replicate one, this is another replicate, that's another replicate, that's another replicate, because the application was done once. Yeah. Okay. So why do we need to replicate? So the first thing is that replication permits the estimation of experimental error variance. I believe now, at least you should have done one statistic course and you know what to call a t-test. When you're calculating, we're doing what called t-calculated. We always have this formula here. If you're comparing two groups, we can talk about X bar A minus X bar B divide by the standard error of X bar A minus X bar B. Okay. So remember, this is the difference between the treatment, and we say we need unbiased estimate. And this is the estimate of our experimental error, which is the noise. Now, of course, we want to, after this, we are going to compare T that is calculated versus T from the table. And we all know that if T calculated, that's a song that we sing when we are doing the introduction to statistics course. You say if T calculated is greater than T tabulated, you reject the null hypothesis and so on and so forth. Now, and the issue is going to be here. Let me change the color of my pen now. Okay. So the issue is going to be here. This is my noise. And in an experiment, you want to try to control as much as possible this particular noise here. Now, of course, one way of controlling the noise is to have a situation like this, where you have your experimental units are uniform. Okay, when experimental units are uniform, I apply the same treatment, my background noise is highly controlled. So you can see from here, if I have, this is a small example here that, of course, it goes back to, this is our values there. So from the first rat, we got 10, the second, we got 9.5, the third, we got 11, the fourth, we got 9. So if you calculate the variance, this is the variance that you will observe. And this variance here, we call it our experimental error. And we can use it in this to calculate, and then we'll be able to test whether there is actually difference between A and B. On the other hand, if I make this assumption that these experimental errors are uniform, experimental units are uniform, then this variance here, which is much bigger, is the one that I'm going to use under there. And because this is very big, it's likely to make the entire calculation, the value here, to be very small. Now, when T calculated is less than T tabulated, then you're likely to fail to reject the null hypothesis. So you will declare that the two treatments that you're comparing are not significantly different. And yet, it could have just been because of the noise that we have there. So the first thing is that if you don't replicate an experiment, you will not be able to calculate the experimental error variance. If you don't have experimental error variance, you are not able to do any statistical test, whether it's a t-test or it is a z-test or it's an f-test, whatever test you're going to do, you're going to have challenges doing that. So the first thing that we need is you need to replicate your experiment. Experiment without replication is no experiment. You need to pay a lot of money to Professor Susan to help you analyze such kind of data. But if you don't have that money, then you better listen to what I'm telling you now. And then you save yourself some money. Maybe you meet me one day and buy me a bottle of beer. Okay. Are we together? Hey, you say, what is pseudo replication? Pseudo means not real. I mean, you're trying to, you're just from the same unit. We want to create more replication from the same experimental unit. The difference between an experimental unit and a sampling unit. Okay, I can go back here. That buy me milk. Okay, so I just want to have here that the experimental unit, as I defined it, is the unit, the smallest unit of the experimental material to which the treatment is applied. So if, for example, we are having an experiment involving comparing fertilizer, different fertilizer level, in most cases, what you do is that you're going to prepare your soil in this, and then you're going to add your fertilizer in there, okay? So this is your experimental unit because this is the unit which receives your treatment. Now, after you planted your crop and it has grown, you're going to come and take measurement. Now, this all group of maize here represents your experimental unit. The entire experimental unit is represented by this. Now, because sometimes you don't have the time or you don't have the resources to take measurement on each and every plan in your experimental unit, you can decide to just select a few of them. And now each unit that can be selected is what we call our sampling unit. Now, if you think you are smart enough, you may decide to say, well, I've just done, let's say after this lecture, you realize that you did not replicate your experiment. So you just go now smartly and then you say, well, this one pot of mine that has nitrogen fertilizer has 5 plants. I'm going to consider each of them as my replicate. So this will become my replicate number 1. The other one is replicate number 2, replicate number 3. That's what I call pseudo replication. They're actually sampling unit. So you're trying to lie or trying to cheat the system that you've actually replicated, but you're not replicated. So that's what we call a pseudo replicate. We are going to see how we measure errors and sources of error in experimental design when we do the analysis of variance. Any other question? Wait, we don't go ahead too far to know now the number of replicate that we have. We have just started talking about replication. We are defining, and then we are going to move on. Okay. And we are not going to say everything here, because if we say everything here, then you'll have nothing to read on your own. So we are going to leave some for you also to work hard. Okay. Okay, so the key point here is, if you're doing an experiment, please make sure you are replicating your experiment. If you don't replicate the experiment, you will not be able to estimate the experimental error variance. There is no variation when you have only one unit, okay? You cannot study variation when you have only one unit. You cannot tell us your girlfriend is very beautiful if you've never seen any other woman. That will not be, you're comparing that in relation to what? So we need replication in order to estimate variation. Variation only exists if there's more than one replication. Okay. So, okay, some questions are going to get answered along the way. Okay. The other role of replication is replication. Increasing the number of replication increases precision. So you replicate in order to increase precision. So precision means how much noise you have, or how certain you are about the estimate that you're going to come up with. So let's say, for example, here we have two different experiments. We have one experiment here we have one experiment that only deals with 4 rats. And then in a second experiment, we have one experiment that deals with 8 different rats. The same. Of course, in one experiment, the number of replicate is equal to 4. In the second experiment, the number of replicate is equal to 8. Okay. So if I looked at this is my second set of experiment. This one compared to the previous one. Of course, you can get the mean of this by adding this plus that plus that plus that and dividing by the total number to get for you the mean. So you can estimate the mean reaction time in this particular experiment. Now that mean reaction time also you need to provide what we call a measure of precision. So a measure of precision is simply the square root of the variance over. So from here you can see that if you measure the variance of each observation as they vary from from the center, the variance of the mean is slightly going to be different. Let me just try to, okay, let me try to stop the presentation a bit. I want to illustrate something. Okay, so let's say, for example, that we in this class here, I'm going to take the height of let's say I have student number 1, student number 2, student number 3, number 4, up to say participant number 300. Okay, so this will be what we call, this would be the population, that is everybody that is in this. Now from this population I can take maybe a sample of size 10. I want 10 people that I will use to estimate the height. So I can take sample 1, 2, 3. Let's say I take let's take just 4 people. So this one has 1.7, 1.6, 1.5, and then 1.8. Okay, so when we are calculating the variance S square, we all know this formula, I think. We say summation of xi minus x bar squared over n minus 1. So this variance here, this variance that we calculate, is we are calculating how each of this value here is varying from the center point here. But we could change the scenario a little bit, and instead of looking at the variation here, we could look at a different kind of scenario. Let's say we get the variance, we get the mean of these 2. So what's the mean of these 2? Is 1.65. The mean of these two, what is the mean of these two? 1.75. Now, if we get another, okay. So I want to ask the question here. Okay, so maybe 1.65 is also 1.65. That's not very good for my comparison. So let me change this to I want to change the color of my pen to red. Let's assume this is 1.6. So then this one become 1.75. Okay. So I want to ask, does the mean of the sample also vary? Does the mean of the sample also vary from what you have there? Does it vary? Because one is 1.65, one is 1.75. Okay. Yes, it varies. Now the question is, does it vary more than the variation in a single observation or less? Does it vary more or less? Less. Regina say less. Andrew say less. Okay, somebody still telling us this is 0, which is okay. Okay. Okay, so somebody said, okay, so I'm going to change, make a little I'm going to make a change a bit again. And then I want to change this value here. So I want to change this to 0.2. Okay. So when I change this to 0.2, what will be the value here now? Will be 1.3. Okay. So when we are calculating the variation here between single observation, we are calculating the variation of each of these from the mean. But when we are calculating for the sample mean here, you see in this particular case, what is happening is that you see in this set here, one value is 1.2, one value is 1.8. So 1.2 and 1.8, when you bring them here, you combine them and you get the mean, they make one particular value, 1.3. Now, so there's a variation between these two values. In the single, in this formula here, that variation is going to be reduced because they are now being represented by one single value. So you can see two values that are extreme. One is 1.2 at the shorter end, and one is 1.8 at the lower end. So the variance of a single observation is always higher than the variance of the mean. So, and the variance of the mean, which we call the variance of X bar, is given as S square, which is the variance of a single observation, divided by the number of replicate the sample size. That's the number of observation that you use for calculating the mean. So you can see from here is that the upper parts are the same, but then this one is divided by n. So in that case, the variance of the mean, sample mean, will always be less than the variance of individual observation. So that is what is happening here, that in this case here, if we use 8 different observations, the variance of the mean is going to be S square divided by 8. Here, the variance is going to be S square divided by 4. So even if we have the same variance, if you have more replication, you get a smaller variance, and a smaller variance mean higher precision. So you can increase precision of your estimate by increasing the number of replications. So one reason as to why we do replication is to increase the number to increase the level of precision of your estimate. So somebody wanted to know how many replications. We're going to answer that. The other reason as to why we do replication is that replication in an experiment will act as a form of insurance. So if you don't replicate, a goat comes running around and eats part of your experiment, you'll be done. So if you have 4 replications, if a goat eat one plot, you still have the 3. So that replication would have saved you. The other reason as to why we replicate is that the replication increases the range of validity of your results. So you can compare your results under different conditions, and then whatever conclusion that you come will have a wider application. So look here, this cartoon here is telling us that we have 2 people who are testing the depth of the river. For one person, the river is very deep because it's short. For the other one, the river is shallow because it's tall. So if you do your comparison under this condition, you do your comparison under a condition here, you should then be able to come up with a kind of inference that apply to short people and inference that apply to long people, to tall people as well. So it increases the range of validity of your results. So this will, so in those who do what we call multi-environment experiment, you observe your result over large samples of farms, and from that farm you could have different soil, you could have different management, you could have all sorts of things. So at the end of the day, we can say a particular variety is stable because it has been subjected to several conditions, and that the several conditions can come in forms of replication. So the number of replicate in this case, if you're going to replicate in order to increase the validity of your results, will depend then on the context in which you're doing your experiment and how widely do you want to test something to be convinced of the result. So those who do multi-location environment will say, well, you need a minimum number of environment before your result can be considered to be valid. The other question, how should replication be done? I already talked about pseudo replication. The next thing that I want to talk about is what we call biological versus technical replication. So this one mainly people who do things in the lab. So I can pick a leaf sample from one plant A and then a leaf sample from plant B. I go to the lab, I make one solution for A, I make one solution for B. Now to run, I can divide this solution here into 2 or 3 test tubes and run them. Now, because A1 and A2 came from 1, I cannot say this one is a biological. These are what we call technical replicate in the lab. So they're equivalent to what we call pseudo replication. So at this level here, you have 2 technical replication for A, you have 2 technical replication for B, but you only have 1 biological sample. You have 2 biological replicate, because this one come from different plants or from different fruit. So when you're doing the analysis, the issue of replication applies here and not here. The one here is mainly to see the repeatability of your experiment, of your results in the lab. So when you're going to do the analysis, then it has to come to the biological replication, unless you're analyzing to see whether there are differences in technical replication. Then the kind of question that you're trying to answer there is going to be different from the one that somebody's interested in answering using biological replication. So how to determine the number of replication that ones need to require? So the first thing is, as I talked about, there are two sides to a design of experiment. There's the issue of experimental material, and then there's the issue of the treatment that you're going to compare. So here, if we look at, if we have experimental material, say there are cows here, there are also cows here. So would you expect to have similar number of replication in this, and then in each of the 2 cases? So here, for example, you see that the animals here are more uniform. There seems to be similar age, there seems to be similar breed, or this, although this looks different, the conditions appear to be similar. Now here also we seems to be see some differences here. The breeds also seems to be similar, but we seem to have different ages and all that. Now, if I'm doing an experiment in this case here, where I see my experimental material is a lot variable, I would need a lot of replication to capture the situation here, compared to somebody here who is doing an experiment using this kind of material. So the pattern and magnitude of variability among the study subject or the study material will determine how many replications you'll actually want to have. So in this case, if there's a lot more variability in my material, I will have more replication compared to if there's less variation in your material. And I see if there are some questions. Okay, later. The second factor to determine the number of replication is the size of the difference to be detected. Now, let's say, for example, I have a new variety. So let's say variety A. Okay, so variety A, let's say it gives me, when planted in 1 acre, let's say you get 800 kilograms. And then variety B, when planted, you get 100 kilograms. Okay, so the difference that I'm trying to detect here is 700 kilograms. Okay, so this is our condition A. Okay, so let's look at another condition here. Let's say I'm comparing two animals. Let's say I'm comparing 2 breeds of animal for milk production. I have now I'm trying to draw a cow, but I already started very badly, so I'm not going to continue. So let me try to wrap now. Somebody has laughed at me, so I'm going to draw now. Okay. So let's say we have two breeds of animals. Okay. Ellen, don't laugh at my cow. He said looks like a bacteria. Okay, so I have two breeds of animals here. Okay, this one gives me 30 liters of milk. This one's give me 28 liters of milk. Let's say 29 liters of milk. Okay, so the difference I'm trying to detect here is 1 liter. Okay, so now the question is, which of these two experiments do I have very many replications to achieve, to test my results? I want to see, is it where I'm getting the meal experiment? I need more replication in the meal experiment. Why? Okay. So it seems to be agreement between two people that the meal experiment will need more replication to take, because what I'm trying to, the difference that I'm trying to detect here is a lot smaller compared to the difference that I'm trying to detect here. So with this very small difference to be detected, I need a lot of replication so that I'm able to even out the noise. Remember, a noise is likely to affect this experiment very, very, very much. So if I come back to what we talked about here, the role of experiment replication in increasing precision, then you see from here that I'm actually better. In this case, for me to remove the effect of any noise, I probably need a lot more replication. So here, the size of the difference to be detected, the smaller the difference between the treatment to be detected, the higher the number of replication that will be required, okay? Like here, I said, if you want to compare the noise made by rat and the noise made by lion, you probably don't need to do an experiment. You have zero replication, you're still going to come to the same conclusion. But if you're comparing two rats, the amount of the noise made by the two rats, you're likely to need a lot of replication, okay? Of course, there is the required significance. So if I want to be 99% confident of my results, I will need more replication compared to somebody who wants to be 50% confident of their results. Okay, and this one doesn't care whether the difference is there or not. And of course, there is the issue of resources that are available. So here you can see that if you have, most experiments require resources, you need money. So even if you want to have very many replications, but the greenbacks may be a limiting factor. So you may reduce the number of replication as to fit in what you have in your pocket. The other principle of experimental design is what we call blocking or restrictions. So blocking control or minimizes what we call non-treatment in variation in an experiment. And we already saw that there's a possibility what we call treatment variation can confound the effect of the treatment. It can increase the noise in an experiment and makes it very difficult to detect any differences that we're looking at. So here, for example, we are comparing, you see the noise here compared to the noise here. So here, because the dogs are different in sizes, the differences in size of the dog actually is reflected in the high amount of noise. And then because of the high amount of noise, we may not be able to detect any differences in the treatment unless we bring in what we call blocking, okay? So we started with this picture and then somebody saying, well, there's the issue with shading. So you can see from here that the plots that are actually closer to the trees appear to be more shaded compared to the plots that are far away from the trees compared to these plots here. So if I'm not going to take into consideration this observation here, let's say I'm comparing this plot here with another plot here. Okay, I'm comparing this one here. Okay, so there's going to be a problem here that this plot is shaded, this is not shaded. So the comparison here is, first of all, there's going to be confounding. If I'm going to observe a difference here between this and that, I'm not going to tell whether this difference because of the shading versus no shading, or whether this effect is because this variety here performs better than that variety here. So that's what we call shading of that. But that shading effect also can now go and contribute and become part of the noise that I have in my experiment. So let me ask the question. Let me make another drawing here. So let's say we have another plot here. This is treatment A. This is treatment B. This is also another B. Okay. Now, which one gives me a better comparison? This, or let me put another color. Or this. Which one will give me a better comparison? I want to see in the chart. Yeah, it's a and B in the shade. So A and B in the shade will give me a better comparison than A in the shade and B outside the shade. So this will give me, so this is where the issue of blocking comes in then. So with the blocking then, what we say is that, let's create a condition where we have uniformity within the block. So we're going to define what a block is. So blocking can allow us to remove or control the effect of variation in the experimental material. We can take care of the non-treatment variation, as I talked about. There are other ways of also controlling non-treatment variation. One of the ways is to do what we call homogeneous material, use homogeneous material, or use homogeneous management, and then use careful measurement. So this is when it comes to taking care of your experimentation. So what is a block? A block is just a group of experimental units that are expected to be similar. So a block could be plots with similar soil type, same breed of animals. So if I'm doing an experiment in animal science, I would consider this to be a block, because within here, my experimental unit, which are the individual dogs, are similar. This can be another block. This can be another block. And this can be another block. Although the difference between block 3 and block 4 may not be that much, but you can actually see that there may be a big difference between block 2 and block 4, block 2 and block 1, block 2 and block 1, and then you have that. So this is the simplest. One can define a block as simply a group of experimental units that are similar. And the similarity here is going to be based on what you think is likely to affect our experimental, the outcome of our experiment, or affects our treatment. In the field, you may look at something like this. So this is the reason as to why we call everything why we talk about blocking in experimental design, because they started from field experimentation. So this can be a nice block. Or it could also be all of this as a block. Okay. And then, of course, the third principle, so experimental design, what called replication, randomization rather, sorry. So randomization is simply the process of assigning treatment to the different experimental units by chance. So like if I go back to my dogs here, for example, first clean it up. So let's say I have here, so I have my 1, 2, 3, 4, 5. So let's say I have treatment A, treatment B, treatment C, treatment D, and then treatment E. Okay, so I've decided here I have 5 treatments. I now know how many replicates I want to have. I know what I want to use for blocking. The next thing then is say, okay, now I want to assign treatment now to these animals within my block. How do I do it? And this is where the process of randomization comes in. So here I need to look for a way that will ensure that A has an equal chance of falling on any of these dogs as my experimental unit. Okay, so each treatment has an equal chance of ending up in a given experimental unit or subjects. And then we always say this is an explicit process and it's not just a mixed up. Those who do clinical trials, you first get a group of healthy adults like most of you, and then you can randomly assign them to 2 groups. There are groups that will receive the treatment, and then there are groups that will receive the placebo or the control, and then you go ahead and do your follow-up after you've done. So the random assignment can actually take place there. So within a block here, so this is, for example, your block 1, we have plot 1, 2, 3, 4. So you can decide which plot within a block actually receive which treatment. So you can do your randomization process here, you do your randomization process there. So why do we do randomization? The first reason why we do randomization is that we want to avoid favoring some treatments. So treatment, you can favor some treatment either consciously or unconsciously. So sometimes you decide and say, well, the plots that are nearest to the road should get special treatment because that's what the plot that many people would see. So you can, in one or the other, try to have that. But also one of the reasons why we do randomization is that the statistical tests that we do, like the F test or the T test, are based on the assumptions that the treatments are randomly assigned to the different experimental units. Otherwise, then if you don't randomize, then you cannot do analysis of variance because it's simply the assumptions does not meet what you actually are talking about. The other reason why we do randomization is we want to avoid some unknown systematic variation that may exist in your material. So here, you may have your plot of land nicely with a different, this is a piece of land on which you want to do your experiment. And on each of these, you have your plots. But then unknown to you, there's actually variation. These different colors actually show different variation. So if, for example, you decide to say, well, I think if this is my, let's say this is my block, my plot 1, plot 2, plot 3, plot 4, plot 5, and plot 6, then say, let me do like this. All my A should fall on the first line. So A will be here, A will be there, A will be there, like, and so on. And then, so by the time you reach D, D could be falling somewhere here like this. So you see now, you've actually landed yourself in trouble because of you did not do the randomization process. So you can find yourself completely into a problem. So there is actually a variation that exists that you probably don't know. Or if you look at this, for example, you can say, well, A falls there, A falls there, and then A also falls there. So somehow by chance, you could actually get yourself into some kind of problem. So randomization ensure that your treatment can fall anywhere within the system here. Okay, so maybe we get a small break before we come and continue with the design now. We're going to look at the different design now. Can we have a 10 minutes? So let's have a 10 minutes break and then we come back.
Societal leadership.
Ce soir, nous avons l'heureux plaisir de lancer officiellement la triennale de notre réseau commun RUFORUM.
Today, we meet at a consequential time in Africa's evolution. Across the spectrum of societal leadership, public, business, academia, civil society, we need to recognize the crucial importance of science, technology, and innovation, and commit ourselves to the cause of excellence in science, technology for sustainable development. We are literally in a race against time.
In Africa, where the youth population comprise more than 60%, efforts should be made to prepare them for the challenges and full participation in the national and global economy.
You are the experts in this field, and we are waiting your recommendations and action plans that will come up with from this conference, especially after the COP26 conference.
Bienvenue au Bénin, plein succès à vous. Vive le partenariat au développement de nos États. Vive le forum, vive nos universités. Je vous remercie.
In spite of COVID-19 challenges, the first RUFORUM Triennial Conference, the 16th Annual General Meeting, and the 7th African Higher Education Week brought together policymakers, including ministers and technical experts, higher education leaders in Africa, development partners, private sector leaders, researchers, innovators, students, and farmer organizations, among others. Over 1,000 delegates from across the world attended the blended event.
This event builds on various convenings that the forum has had, virtually and face-to-face, including the just concluded United Nations Food System Summit. We had a meeting of African heads of states to discuss how African governments can work together with universities to support agricultural transformation.
The Roof Forum has demonstrated investments in human capacity development, in training our young people into research, building new platforms for knowledge exchange, both old and young, in different parts of the continent, assisting our governments to formulate policies. But having said all that, I think it's the time for us to take stock and start asking ourselves the hard questions. How have we fared with our commitments? And so I call it a time of action.
With the theme of operationalizing higher education for innovation, industrialization, inclusion, and sustainable economic development in Africa, a call for action. The meeting was the next step in Roof Forum's journey to create a more prosperous Africa.
It is basically a call to action. Both the private sector and the policymakers are part of this dialogue, in addition to us, our own individual universities and our global partners.
Many of the Roof Forum family had not traveled since the start of the pandemic, and the emotion and relief of the face-to-face reunion was palpable.
You need to be able to connect with everybody, to talk, because you can't take that away, the face-to-face, the physical, talking to somebody and being able to just connect is very important, yes.
The event was also the first of its kind in a French-speaking country.
We are in West Africa, we are in East Africa, we are in Central Africa, we are in Southern Africa. So we have covered the entire African region.
Putting them together, put them together to think as a whole for the whole continent.
Well, definitely this is the Africa that we are looking for. The Africa where we come together.
There was a strong sense of female leadership at the meeting.
المرأة لو حصل لها تقوية لمقدراتها هي اللي بتقدر تقود التنمية في أي مكان. فدي الحاجة اللي بنتمناها في أفريقيا.
The new board chair was elected.
Cameroon is in Central Africa. I would like to see more participation from universities in Central Africa.
Entrepreneurship and private sector linkages as tools for young Africans' participation in development were a major focus.
And issues of industrialization, commercialization, entrepreneurship, innovation are all very critical. And I think what we're being challenged to do now is say, yes, we've offered programs, we've offered degrees, but what can make a difference is action. And it's about the people we serve. It's about those rural farmers, rural families that will never go to a conference. After the discussions this week, let's go back and work.
The digital revolution took center stage. E-learning deliberations emphasized the challenges and opportunities presented by the move to online education. Roof Forum's e-learning platform was launched.
Roof Forum is going to lead the African integration in education. The time has come to lead the delivery of programs across Africa by tapping into the expertise they have built.
I do appreciate that the Roof Forum Triennial Conference brings together eminent persons and relevant stakeholders that have the capacity to drive Africa to the Africa we want by 2063.
L'Afrique que je veux est une Afrique unique, une Afrique solidaire, une Afrique de développement, une Afrique qui cherche ses solutions en son sein.
The mission and the vision of Roof Forum is actually the future of Africa. It's priceless, and we're very grateful for that.
Hello. Okay, so I'm back. Welcome back, everybody. I want to start by answering a few questions. So I want to start with Doris. Is saying I'm working with weeding rats. I observed that most of them do not eat unless they eat together. I decide to put the food together and mark the animal. Okay, pseudo replication explain. Yes. Okay. Yeah. What you said simply done is, like you said, you put the animals and then you mark them for the different. That certainly is pseudo replication. What you should have done would be to create different cages, and then the replicate in this case is not, you're not counting within the cage. You could have a cage where you have the different rats in as your replicate. So then I can have maybe cage one where I apply treatment A and I have more than one rat inside there. So then I have the cage as the replicate, and then the individual rats as your sampling units, which is the pseudo replication. Yeah, so those are the things that sometimes you learn to appreciate when you do the experiment yourself. That if you are to give the rats each one independent the food, then they will not eat. So that one is there. So I think that is answered. The other one is, how do you analyze data with unbalanced replicate? For example, we have not reached the analysis yet, so we'll reach there. What happens when it is observed that one experimental unit group animal or basal diet group consume only less than half of the basal diet each day? Would the difference in feed consumption be assumed bias? What can someone do to ensure that the least feed consumption is comparable? Yeah, so probably you could also look at your you record probably the amount of feed that is eaten in each cage, and that we could also use that as a covariate in the analysis later on. But I certainly see you're having real experimental challenges that actually people have. So you need to start thinking along the way, how do I handle situations like that? So yeah, the size may actually come in. If we get precision by increasing replication, what can we do in clinical trial where patient might be scarred? Yeah, of course, that's why you may have as many replicators as you want, but the reality may be different. Yeah, so in this case, if the patients are not there, then mean you cannot have the number of replicate that you want, and your precision may be low. So the question is, should you still go ahead with the experiments, or because you could also waste your resources and come up with very low precision that will actually not work. How does one determine the size of the difference in experiment that has not yet been carried out? How do we estimate the size of the difference? Well, sometimes you may not have to know the size of the difference that's there, but you may also be able to, you may also be interested in, for example, knowing what is that difference that you think will be of significance in practical terms. So if, for example, you're dealing with the drugs that you have not new drugs that you're comparing two or three different drugs. So probably there should be a clinically important level like that. You say, oh, if recovery cannot take place within, if it's painkiller, if it is going to take a full day without you getting relief, then it may not actually be good. So sometimes there may be a challenge in determining the actual size of the difference that you're expecting. But as I indicated before, we expect that somebody doing an experiment, you should have some idea of what you're studying. So you can go into literature and then, but you can also look at what called the significant, what makes sense in practical significance. I think it's the same question with the clip that if there's assumption that we have prior information on the magnitude of the difference, what happens if the assumption is not available, maybe prior checked, or Yeah, so that is where you need to still get some kind of information that you actually need. So maybe I said, well, a difference of if you're, for example, let's say you're comparing a new variety with an old variety, you don't know how much diff is likely to be. You could say, well, according to a standard in some countries, you cannot release a new variety unless you have a 10% difference with the what is existing or the base on the market. So that kind of information can actually be used to solve to answer the question. Does block means having homogeneous population or different? When you're doing survey, for example, we talk about stratification. So stratification is similar to blocking. With stratification, what we do is that we assume that our population is not homogeneous, and therefore we can group that population in two groups within which there's homogeneity, and then between the group there's some differences. So when you're talking about blocking is similar to what we call stratification in sampling survey. So again, Doris say which one comes first when distributing experimental animals to treatment, blocking or randomization? We are going to look at the different types of design. The first design, what we call randomized complete design, there's no blocking in. So in that case, the randomization starts off immediately. But in some other design, the blocking is done, then randomization is done within the block. Okay, good. What does it mean? Does it mean that anytime you open R, you must install? No, you don't have to install all the time. So let's now go back to the presentation. I'll answer other questions later. Now my colleagues will answer some of the questions that has been raised. Okay, so we come up on straight away what we call completely randomized design. So with completely randomized design, the main assumption here is that the experimental units are homogeneous. So like you see in this, these rats here, they're all the same. It's just one rat that I replicated in the picture. So we assume they're homogeneous. So in this case, like the question, just to pick up from the question, someone said which one comes first, randomization or distributing the animal in two blocks. So in this case, there is no blocking. As you can see, all the experimental units are uniform, so we don't need to group them. So we go straight away to the randomization process. And here, the randomization process is done in one go. So we put all these animals together in one basket and decide which one receive any of the treatment that you have. I provided you with some scripts. So this is one of the scripts that is used for randomization in CRD. So what happens here is then that once we have our rats, we can number them from number 1 up to say number 12. And then we can decide which treatment number 1 gets by using a simple randomization process. So this is just a simple script that you have the script already with you. It's shared with the material for today. So here, the first thing that we need to do here is we set what we call a seed. So a seed allows you to get the same randomization whenever you do it. Once you set the seed, if all of us set the seed to 500, we should all get the same randomization. Otherwise, the randomization will be different. So what we are going to do in this case is that we are going to create an object, which we are going to call F. I think Ellen and Susan talked a lot about objects yesterday. So I think I can use them with a lot of ease now because they've already been talked about. So I'm going to create an object called F, and this object here, it will be converted into a factor. We are going to talk about factor later on when we're doing analysis. So here, what we're telling R to do is that you pick this object here, and then replicate, each of them should be replicated 4 times. So what you're going to end up with, you're going to end up with something called F, and F is simply going to be equal to something like this. You have A, A 4 times. And you have B 4 times. And remember here, each of them is, each treatment is replicated 4 times. So we are creating an object which has the size of 12. And then we're saying, okay, now what we do, we have this object here, A12. So this is our treatment that we want to apply. So we use now this simple sample. That means go to these 12 objects that I have and distribute them to the 12 experimental unit that we have. Okay, so here it says sample from this and distribute among the 12 experimental unit. And then here you sample without replacement. So if I already picked A and assign it to rat number 1, the next assignment is going to be now having only 3 A's. And then you assign the one to rat number 2, you assign to rat number 3. So in this case, the first rat is given treatment B, the second rat is given treatment C, the third rat is given treatment A, the fourth one is B, the fifth one is A, the sixth one is C, the seventh one is C, and so on until the 12th one also get A. So this is what you can do once you set your experimental material ready, all your experimental units are ready, you can use R to do the randomization process. We're going to run this tomorrow, maybe early in the morning if we don't have time to run it today. But I actually don't even need to run it with you because you can easily run. So this is now finally the results of the assignment, okay? So the assignment is done in one go. So all the experimental units are assumed to be similar with respect to characteristics that's likely to affect the response. This is commonly used in the laboratory in a screenhouse experiment, but in the field, in a culture field, this has a rare application. So it's less likely to move there, okay? Of course, the main objection to the use of CRRD, especially in the field, is that it's not very efficient. Okay, because the randomization is unrestricted, so the experimental error include the entire variation in the experimental unit except the treatment. So let me change the color a little bit so that I can put some emphasis on the, okay. So here, remember this is our background noise. And because of the assumption that the experimental units are uniform, that means any other variation that exists between the different experimental units are all going to lump up together as part of our experimental error. And in that case, you're going to have a very high noise in your experiment, and you are less likely to achieve your objective of testing for the different treatment that you have. I already showed you that if we use the similar experimental unit, we get this kind of variation. But we use different experimental unit, we get a higher amount of variation than what we'll have. So if the experimental units are not homogeneous and you assume they're homogeneous, you're going to end up with a very high amount of noise, and that is going to interfere with your ability to detect differences between the treatment. So the next design is what called randomized complete block design. So with randomized complete block design, we all know that experimental materials are always not similar, are not homogeneous. So if, for example, you're an animal scientist, so I mean, this could be, if you're doing an experiment in the farmer's field, the farmers can give you any of these animals as your experimental material. So you can have a situation where the animals are fairly uniform. You can have this situation. you can have this situation, you can have this. So it's really, really important that you understand the experimental material that you're going to use in your experiment in order to be able to design an experiment that is efficient, okay? So experimental materials are always not homogeneous. So assumption of this, of completely randomized design, is likely not going to work. And if you insist on that, you're likely to get a lot of noise in your experiment and you will not be able to detect any differences between observations, okay? So here are a few assumptions that there is a predictable trend in variability in the experimental material. So you can, I mean, like from here, I can see the trend. I can see this animal here is emaciated. This one is of a different breed. This is looking, this one has a very good score condition. So the pattern should be, you should be able to predict the trend in variability of your experimental material. And then you should then use the variability in your experimental material to group the experimental material into blocks. And when we say within the block, there has to be homogeneity, and then there could be differences between the block. So I still want to emphasize this, that the knowledge of your experimental material is very, very important as far as that is concerned, okay? So this can be your block number 1, this can be your block number 2, this can be your block number 3, and that's your block number 3. So in this case here, as Doris asked, in this case, the randomization process will come after first grouping your experimental material. Randomization now will come because you're going to apply your treatment within the block by block. In the field, you could have something like this. As you can see, as you move down here, the fields get a lot darker in the color. So this could indicate, for example, fertility level. So here you have block 1 could have low fertility, and then the last block number 6 could be a block with high fertility. So this could also be similar to the shading experiment. So this could be high shading and this could be low shading as you move around. So if you go back to our shading experiment, let's grab it off. Okay, so you can see from here that we can actually draw this as our, maybe we draw this as our block. So this is our block number 1 with a deeper shading color. Then this could be our block number 2. And then our block number 3. And then within the block now, that's where you do your randomization process. When you already have a block, because within the block, we are expecting uniformity within the block. Okay, so we can do the same thing. This is another way that another way of doing the randomization. So the first thing is therefore this is called randomized complete block design. That means that each block is complete. By complete means if you have 5 treatments, then all the 5 treatments should appear in each block. Otherwise, then the block will become incomplete. So here, I have 5 treatments, A, B, C, D, 6 treatments, A, B, C, D, E, and F. And then I want to come and every time I want to come and see how do I distribute this within my block. So this is our block 1. So I get my 6 treatment, I sample without replacement 6 times. So the first one will assign to plot number 1. The second one will assign to plot number 2, the third one to plot number 3 until plot number 6. Once I'm done with randomization in block 1, I go do randomization in block number 2, randomization in block number 3, in block number 4, in block number 5, in block number 6. Once I'm done with that, I come and combine them up into some kind of, yesterday they talk about a data frame. or a matrix. So we can combine this one up into a data frame, and then we create a different component. I'm going to talk about this when I come to running the R script. So at the end of the day, we are going to create something called a plan, and the plan is what you actually now use in the field for that. So based on what we've run here, this would be what will be in the block number 1. So in block number 1, the first plot gets treatment C, the second one gets treatment A, the third one gets treatment D, the fourth one B, the fifth E, and then the 7th gets treatment F. And then once we are done with that, we go and do randomization in that, we go do randomization in that until when the whole thing is done. But this one is just a rough way of doing it. If you go to, there's a very, very nice package called Agricole in R. So you first need to install the package Agricole. I will ask you to install package Agricole today, those who don't have, and then so that tomorrow or maybe in the after this, we can talk about it. So once you install this package, what you need to do, you need to define your treatment, the set. In this case, you define it as 1, 2, 3, 4, 5, similar to what we define here. This one here. This one we define it as A, B, C, D, E, and F. And then we use now this small function here. Okay, so this small function here will get your treatment here, which you call treat, and then we'll assign to the different plots within a block, and then we'll repeat for each of the block, and then you create an object called out design. So from the out design, we can pick one component. Yesterday, Ellen, yesterday and the day before, Ellen talked about using the $sign to extract a component of an object. So here we can extract what is called a book from this design here, and then we can manipulate that object properly so that we can have one column that has all the blocks in. You see now we are looking at the levels of this particular block, and then we can assign this. So this is what we'll find and come up with. So we are saying here is that plot 1 in block 1 will have treatment D. Plot 2 in block 1 will have treatment F, and so on and so forth. So this one we are going to look at it practically. You'll run and you will appreciate what I'm talking about here. So after this, once you have your experiment, you can go to R and you can use this in order just to design your field layout. You go to the field and do your experimentation without any problem. The other design is what we call the Latin square design. So with the Latin square design, in the case of randomized complete block design, we were using only one factor to do the blocking. Like in this case, we use the shading. So there's only one direction for blocking. So assuming now that there are two factors that we need to block against. So here we have, you could have this going out like that. But you could also have a situation where there's another factor going on in that case. So you can see you have blocking in this direction. You could also have blocking in this particular direction. So you can block in two different directions. So in the field, for example, you could block using soil fertility. You could also block using the gradients that you have. So as you can see here, the assumption is that the fertility gradients and the slope gradients are perpendicular to each other. So I can have block 1, but the block 1 here is in respect to the gradient. So this is at the lower gradients. This would be somewhere again This would be somewhere again, and this would be at the upper gradient. So you can see this is your block 1. This would be your block number 2. This will be your block number 3. And this will be your block number 4. Okay. Now, when you're blocking in relation to the slope, but you can also block in relation to the fertility gradient. Now, with respect to fertility gradient, then you have the line of the blocking now moves in a different direction. So this will be your block number 1. This will be block number 2. This will be your block number 3. This will be your block number 4. Okay, so whichever direction you look at, you have a randomized complete block design. So you can have RCBD here, randomized complete block design. You can also have RCBD there. Okay, so this is more like to just look at it more from visually how it looks like. So in the case of our tree shading experiment, you could also have the shading getting intense along this way, but then you also have fertility getting intense along a certain direction as you have. So you have 2 factors that you have. This is also used a lot in the lab. For example, you can have operator. Let's say this is one operator called James. This is another operator called Jane. This is John. And this is Muna, for example. You can have this as your operator. And then you have machine 1, machine 2, machine 3, machine 4. So you can see that this operator here operate the 4 machines. Okay, so it's a complete block in this case, that all these readings are coming from operator number 1. And all these ones here are coming from operator number 2. These ones are coming from operator number 3. And then these are coming from operator number 4. Okay. So as far as operator is concerned, you have randomized complete block design. You're blocking block 1, all the machine reading from operator number 1, from block 2, operator number 2, operator number 3, operator number 4. But you can also block using the machine. Okay. So all these ones here are for machine 1, machine 2, machine 3, and then machine number 4. So as you can see, these are what we call, these are our columns, and then these are our rows. These are the rows. So that's why Latin square is called row column design. You have your rows and you have your column that you have. A nice example is in animal science. For example, so let's say we have this 4 animals, and then we have 4 different drugs, or the same drug but different doses. There's the control where there's no drug, and then there's where there's a low dose, there's a medium dose, and then there's a high dose. So you can see that animal 1 received in week 1 was not treated with given a placebo. In week 2 was given a low dose. In week 3 was given a medium dose. In week 4 was given a high dose. So based on this, we can block using the animal. So block 1 is on animal 1. That is the first row. Then you can block with animal number 2. You can block with animal number 3. You can block with animal number 4. So this is the row, each block. So each of these is a complete block. Remember, all the treatments are in the block, therefore the block is complete. Now we can also block with the time of application. So remember, you can only apply 1 treatment to an animal at one time. So in week 1, animal 1 received the control, animal 2 received low dose, animal 3 received medium, animal 4 received high dose. So this is a nice Latin square design for animal science experiment, whether for drug test or for nutrition. So Latin square is laid out in row column design. So each treatment occur once and only once in each row and in each column. So here is a special case where the number of replication is actually equal to the number of rows, equals to the number of columns, and equals to the number of treatment. And that there is also where the trouble is going to come. So that means if I have 10 treatments, then I need to replicate it, each of them also 10 times, because that's what the Latin square will dictate. So the total number of plots is the square of the number of treatments. So plot here or number of experimental unit will be equal to the square of the number of treatment. So if I'm having 10 drugs to test, then I need in total, I need 100 experimental units to have. So here you can block in 2 perpendicular directions if it is in the field. And we expect that here you can reduce a lot more variation compared to if you use randomized complete block design. The limitation, the square requirement is actually a limitation in that, remember you see if I have t equals to 10, then I also need to have 10 replications. So the number of replications should also be 10. And the number of experimental unit that I need will be 10 squared. So if these are plots of land, you need 100 plots, and depends on the size of the plot, it can actually be very big, as you see what we call incomplete block design. Now, if you're dealing with smaller experiment, if you have only 3 treatments or you have only 4 treatments, then you will have problem with efficiency, because you have fewer degrees of freedom compared to other design. I'm going to talk about this when it comes to analysis of randomized complete block design. So there are several incomplete Latin square design that are there that can be used if at all you cannot afford the issue, the entire Latin square. You can use incomplete Latin square. So the rows may be fewer than the number of columns in that particular case. So that's what we call, that's our Latin square design. We are going to, you have the codes here, and you don't need any data set or what you simply need, you just go and run those codes and you will get these results here. Okay, so the first thing that I've done here is to create an object called nutrition, where it has nutrition 1, nutrition 2, nutrition 3, nutrition 4, and nutrition 5. Okay, and then you can use design.Latin square, that is LSD. You put in what you want to, the treatment that you have, and then you put in the seed. I'm going to talk about more about the seed when we are running this practically. And then once you've created another object here called out design, and then you can remove something out of it using the $sign. You can get the book and you can label your book, and then you can print the book. So this one we are going to run it. So this is how it will look like. So then I can say in week 1, animal 1 is assigned to nutrition 1. Is assigned to nutrition 1. In week 1, animal 2 is assigned to nutrition 2, and so on and so forth. So this is how the whole thing will move on. So ARA has these facilities that can actually help you to design your experiment and before you can go to the field to do the actual work. And then the other standard design that we are going to talk about is what we call the Latin square design. Not the split plot design, sorry. So the split plot design is used with a factorial treatment structure. So maybe before we go, can somebody help us to define what a factorial experiment is? Can I see in the chat? What is an what is an experimental, what is a factorial experiment? Somebody say combination of treatments. I need to see some answers. An experiment where more than one factor is being considered, yes. More, an experiment whose design consists of two or more factors. But what is a factor? An experiment in which more than one factor, an experiment whose design consists of two or more. Okay, I think we have the people who have responded have some idea what we are talking about. So if, for example, let's say, for example, here, let's say we have tomato, we have three tomato varieties. Tomato variety 1, tomato variety 2, and then tomato variety 3. We have 3 different varieties of tomatoes that you want to test. And then we also have pesticide. We have 2 types of pesticide. Pesticide 1, type 1, and pesticide type 2, okay? Now, of course, variety 1 and variety 1, variety 2, and variety 3 are all related. They're all varieties of tomatoes. This one, it becomes a factor. So a factor is simply a group of treatment that are related. So this is our factor 1, which we can call that factor tomato variety. And this is our factor number 2, which we call it a pesticide. They're all different types of pesticide. So a factorial experiment is an experiment in which 2 or more factors each at 2 or more levels are studied. So here we have what we call a 3×2 factorial. A 3×2 factorial. So if a split plot is only used when you have this kind of study where you have 2 or more factors are being studied together. So in this case, one of the factors will probably require a larger area for application than the other. So between pesticide application and the different types of variety, which one do you think will require a wider area of application? You're saying treatment. Oh, the tomato variety, yes, somebody say tomato variety, another one? Variety, pesticide. Why would a variety, tomato variety, why would a variety needs, okay, tomato variety. Okay, so can you defend those one variety? Okay, somebody say because variety has 3 levels. Okay, Olusegun, can you defend why pesticide need a wider area of application? Are you related to Obasanjo? Some will say because the number of pesticide, because it require a large area of application. If you're going to spray, for example, if you're going to spray, let's say you're going to spray with a tractor or spray with a boom sprayer, do you need a smaller area or a wider area? Okay, I'm going to explain that. But we have some idea that one of the application may actually need a wider area. So it can be used with the CRD, RCVD, or with Latin square. It is more of kind of how the treatments are arranged over the other design. So it's actually not, some people think it is not a design per se, it's just the treatment arrangement. And so what it involves splitting a larger experimental unit onto which the first factor is applied into a smaller group. But I think I will have, let's have this. Okay, so I think this one can probably give us a better argument over which one of the two would require a wider area of application. So let's say you have a tray. This is, you have tray A, and then you have tray B, okay? Now, is it possible for me to spray just this line here with one chemical and leave the rest? I want to see now. How can I control my spraying in such a way that I can only spray one line? Okay, so that's where now the logistics becomes a problem there. It would be easier for me to actually to spray everything here at once, okay? But, okay, is it possible for me to plant just one line with a variety? Okay, so which of the two is easier to apply over a smaller area? Is it a variety or the spray? Which one is easy to apply over a smaller area? The variety. So what we can actually do then, I can actually have each line to be a different variety. So if I'm comparing variety, I can have the line 1 for variety 2, variety 3, variety 4, because when it is planting is a lot easier to control that. At least I can put this and that. Then once I have done this, the next thing I can then do, I can then apply the bigger the spray over the entire experimental unit. So in this case, the bigger unit, which is the whole tray, will receive the spray, and then the splitting is done by the different line that is within the tray. So I can have here, for example, spray with A, and then I can have your spray with B. So in the application, I would say it is the spray which is now split. So the first thing is over a bigger area is going to be the spray, and then I can split my spray into lines with a different variety. So that is why I have two factors here. I have the spray, and then I have the variety, and I can combine them together, and I can use a split plot. Okay. So this is another way of looking at it. So I can get my plot of land and divide it into two. One I spray, one I don't spray. And then within here, I can still break it up into different plots in which I can apply my variety as a treatment. So here I have my block 1, I have my block 2, and then I do the splitting. So this is what we call a split plot. Then I can also split this, then it becomes split plot. You can continue the splitting until you cannot do the splitting anymore, or until when you feel you have all your factors have been covered up. So the splitting can proceed as long as required or as long as it is still feasible. Randomization for the different levels of factor are done separately. You see now the spray and no spray is randomized separately from the variety. So the first randomly assign spraying to the main plot, and then assign variety to a subplot. So what I can do in this case would be, I have two different trays. So this becomes my two bigger experimental unit. I assign here spray with A and spray with B, and then I split the spray into several varieties of tomatoes. So then I am splitting. So the bigger factor, what we call the main plot factor, will have spray, and then the subplot will have the tomato variety. So the implication here is that different experimental errors are used for testing different factors. So the spray no spray uses a different experimental error, and then the variety will use a different experimental error that you have. So we can also use the same package Agricole, and then we can do the same thing and generate for us the design that will decide where the main effect will go, and where the where the main treatment will go, and where the subplot treatment will also go. So this is the same procedure. So then I can answer a few questions, and then we can proceed to incomplete block design, which is the final part of the presentation. There seems to be no urgent questions to answer here. Okay, so maybe the slope gradient can be relatively easy to observe in experimental site, but fertility gradient can be quite random, especially in parts of the field that are used as cow sheds and what. Yes, Wycliffe, you're raising very good observation. So how do we handle that? In most cases, if there are things that we cannot use for blocking, we can introduce them in the analysis by doing what we call analysis of covariate. So analysis of covariate is when you can score the different plots and then use the scores to first correct for the analysis before you can do that. Can Latin square design be likened to feeding different strain of animal, different diet? Well, that is possible if you're not interested in testing the different animals. What kind of design should I use for two factors? First factor with 3 levels and second factor with 3. But that's factorial, so you can use it with RCBD, with CRD, with Latin square, depending on what you have. Please kindly offer clear distinction between Latin square design and factorial. The factorial, I mean, the factorial is about how many factors you have in your experiment and what are the different combinations that you have. A Latin square is how your experimental unit related in terms of grouping what. Can you block them? Somebody wants to, what kind of design should I use? Kindly clear distinction, I think. Can blocking be systematic? Yes. Can we have more example in other fields other than agriculture? Which other field can we talk about? No, it depends on the idea is applicable in all the fields that you have. So, for example, in medical, you can, I mean, you can use male and female as your blocking factor in a trial that you have. Okay, so let me just try to go briefly on and introduce the concept of incomplete block design, and then we see how far we can go. I know people are a bit tired, but we'll see what will happen. So we've just looked at blocking, and we know that blocking can reduce experimental error and provide more precision for treatment comparison. So it increases precision, which is very, very important. And we also emphasize that within a block, there's supposed to be uniformity. Between the block, there's supposed to be, we need the blocks to be as different as possible, but within the block, we need some kind of uniformity that you have. Now, there are times when the number of treatments that you're dealing with are very, very many. So the number of treatment that if the number of treatments are very many, it becomes difficult for you to get experimental units that are homogeneous. Okay, so as the block get larger, the condition become more heterogeneous, and then the purpose for which you've done the blocking may actually fail. So let's say, for example, in this case, we have 9 treatment, and this is, if we have these are our sets of puppies that we do for the experiment. If you only have 9 of, you have 44 of them, that means not all your treatment will be accommodated. So what we could do, we could borrow the neighbor's puppies also. We can see. Now, when you borrow the neighbor's puppies, now the neighbor's puppies don't look like yours exactly. So you have, yeah, well, now you have a block of size 9, but the problem now is within the block now, there is no uniformity anymore. So the idea of bringing blocking was that within the block, you want to create uniformity, but in trying to get a block that is large enough, you're ending up with a block that is actually not homogeneous anymore. So we could decide to say, well, let us have a block that is actually smaller. Instead of having 9, we can have a block of size 4, and this one is a block of size 9. At least within here, there's uniformity. Within here, there's uniformity. But if you combine to increase the size, then you're going to have a problem with that. Sometimes blocks are also based on natural grouping. So for example, litters. And you cannot force a saw to produce as many piglet as your experimental unit would require. Okay. So here, for example, we have 1, 2, 3, 4, 5, 6. And here we have a lot more. So if you have 10 treatment, then this one here is not going to be complete. You only can only test 6 of your treatment here, while here you may actually test all your treatment here. So blocks based on natural grouping of experimental unit may actually have fewer experimental units compared to the number of treatment. So these are realities that we have in the field. So we should then be able to get designs that will help us to handle that. So this will give rise to what we call our incomplete block design. So with the incomplete block design, as I already shown you using those pictures, is that your blocks will have fewer experimental units than you would require to have all your treatment in order for us to have a complete block. So our block ends up being incomplete. So we can classify incomplete block design based on 2 criteria. It can be based on the number of blocking factors. You can have those incomplete block based on 1 blocking factor compared to those based on 2 blocking factors. You can also classify your incomplete block design based on the precision of comparison of the treatment. So here people talk about balanced incomplete block design versus partially balanced incomplete block design. So I'm going to talk briefly about the incomplete block design, and then we'll show you how do you generate the design, and then you should then be able to use that. Okay. So the classification based on random on blocking factors. Remember, we talked about randomized complete block design. So we can have randomized incomplete block design if you're dealing with only 1 blocking factor, and then you can also have randomized complete block design based on 2 factors. You have what we call incomplete Latin square, or we have incomplete row column design. So these are 2 basic classifications that you can always talk about. So if I am to have a summary here, we started with a completely randomized design where we assume that the experimental units are homogeneous. And then we went ahead and said, in case the experimental units are not homogeneous, we can have randomized complete block design, RCPD, where you're blocking with 1 factor. But in case you're blocking with 2 factors, then you can have Latin square design. Okay. Now, if the blocks, if for some reason now you cannot get enough unit within the block to accommodate your treatment, then here you have your randomized incomplete block design, randomized incomplete block design. And then here you can have your incomplete Latin square design. That's what you have based on the classification of the blocking factors. That is what you have. And then the issue of partial versus balanced incomplete block design can actually come within each of these. So you can have balanced incomplete block design is famously called BIP, and then you can have partially balanced incomplete block design called PIP. These are the one, but this can also be under here. Okay, so you decide which one you actually want to use. Somebody talked, I saw some question that I had not answered about the number of having very many treatments and replication. I'll come back to that one at some point. So what is balanced incomplete block design? So with the balanced incomplete block design, each pair of treatment occur equal number of times in the same block. We're going to have a look at the example, and then partially balanced incomplete block design, each pair of treatment do not occur together equal number of times. So we have a block. So if I go back to I like these puppies examples. Okay. So here, for example, like what do I say? So let's say A and B are in this block together. Okay. And then A and B are also in this block together. So you can see A and B occurring 2 times in the same block. We could have C and D here, and then C and D is also here. Okay. We could also have C and D there. So you see C and D is once, twice, 3 times. So in this case, when some combination occur more frequently together than others, that means we are dealing with what called partially balanced incomplete block design. Okay, just a few questions and then we can continue. Does the choice of a design depends on the number of treatment? Yes, it does. And the case is for the case of a split plot, for example, sometimes if you have too many factors of a certain treatment, then you may need to go for a split plot to have that. Can we test 2 treatment type in the same experiment? I don't seem to understand this question very well. Size of block in a split plot. The size of a block, I mean, it's more or less the same as the randomized complete block design. But you remember we said the split plot does not talk about block per se. It imposes itself on any of the design, whether it is CRD, RCBD, or Latin square. Just guys, just hold on a little bit now. I know you're tired, but we're going to move together. Otherwise, you can see even the puppies look happy. One set of the puppies are from African wild dogs, so you don't need to bring them and join because you want to create a large sample, a large block sizes. Okay. So for partially balancing for balancing complete block design, all the treatments are equally replicated. So if all the treatments have 2 replicates, 3 replicates, that is that's it. And then each pair of treatment occur together in the same number of time, occur together in the same block equal number of times. I demonstrated that. So if A and B occur together, okay, there is a question, we say differentiate between a Latin square and a factorial. So the first thing that I want us to appreciate is that when we are talking about the design of the experiment, we're talking about a CRD, we are talking about RCBD, we are talking about Latin square or incomplete block design. We are not mentioning anything in relation to treatment. What we want, in this case, our main concern is what are the characteristics of our experimental unit? Can we group our experimental unit? If yes, can we group them on using one particular criteria? Like, can we group them using the distance from the shading tree? If that is the case, then we are doing randomized complete block design. if we have to do using two factors, and then that will be incomplete, what we call the Latin square design. Now that has nothing to do with the treatment. The treatment structure is mainly then you can either have factorial or non-factorial. You have non-factorial if you are testing only one group of treatment. I'm only testing variety. I'm only testing fertilizer. I'm only testing maybe certain drugs. Now, it is factorial if I'm testing more than two factors. I'm testing drug. I'm also testing gender. That will be what we call a factorial. So when we're talking about a Latin square, we're talking about arrangement of experimental units within our experiment. When you're talking about factorial, you're talking about how do our treatments behave, how do we categorize our treatment, okay? So with balancing complete block design, all pairs are compared with the same precision, even though the differences between the blocks may be large. So at the moment is that all pairs are compared with the same level of precision. The same level of precision because we have the equal number of replications for everything. So remember, the variance of the mean is S squared over R. So if R is the same, that means the precision is the same. That is the whole thing. So when you're dealing with balancing complete block design, you can literally have a balance incomplete block design for any number of replication, any number of treatments, sorry, rather, and any block size. The block size in this case is how many units are there within each block. Remember, in case of our puppies, we have 4 units within each block, so the block size is equal to 4. So, but then what will determine is the treatment, the number of treatment, and the block sizes. The number of treatment and block sizes will determine the number of replication that is required. So if you have 10 treatments and each block of yours can only take 3 treatments, that means for you to achieve a certain number of treatment, you need a lot of replication. For you to receive a certain number of replication, you'll need a lot of blocks in order to capture that. And so the challenge here is that the minimum number of replication required for a balance, to create that balance, complete block design, may actually be too large to be practical. So the minimum number of replication may actually be too large to be practical. You may require maybe 10 replication in order to have a balanced condition. So balancing complete block design are rarely used in field experimentation or even in industries. Then there is a nice example here that I picked from one of the textbook. So here, the experiment was to determine seed germination. So in this case here, we are looking at tomato seeds, and we want to see what factors affect the germination of the seed. So here, the main factor of interest was effect of temperature. Okay, we want to see how temperature affects seed germination. So we selected 4 different temperatures. You can subject the seeds to 25 degrees centigrade, which is the room temperature. You can subject them to 30 degrees centigrade, you can to 35 degrees centigrade or 40 degrees centigrade. Okay. Of course, the thing here is then that the seeds should be in a kind of a control environment chamber. So you can have a chamber that probably looks like this, and then you put your seeds in there, and then you can have this, let's say maybe you can put a number of seeds there, and then, okay, you can have this. Okay, so you can, let's say we control this as 25 degrees centigrade. Okay, then you have another, and then you have another one like that. So in this case, we have 4 treatments. So we expect for each block to have 4 different chambers. So you can have 30 degrees there, you have 35, and then you have your 40 degrees. So in this case, so if this is your first run, okay, so one complete block or one set of replication would require 4 chambers because you are doing this in a growth chamber, but only 3 chambers were available to the scientists. So instead of 4, you have only 3. So that means each time you try to run, you cannot run all your 4 treatment. You can only run 3. So then in this case, your block now becomes incomplete block. So we can run, this is our chamber 1, 2, 3. And the run here in this case now is used as a block. Run 1, run 2, run 3, and run 4. Of course, when you're running this experiment, you expect that the condition at a particular point may be different. Let's say this was run on a Monday, this one is on a Tuesday, this one is on a Wednesday, and this one is on a Thursday. So maybe the one on a Monday was run by Ellen as the technician, and then Susan runs the one on Tuesday, and then I come and run the one on Wednesday. I may come when I'm drunk, and then I can have an effect on, it can have an effect on. So you can use each of us as a block. And then we expect that if I am drunk, then it's going to affect all my treatment that day, will not select any other. Okay, so here then we can look, if we look at the different combination, let's see, 25 and 30. How many times do they occur together? Once here, 25 and 30 are occurring here also once. There's no 25 and 30 here, there's no 25 and 30 there, okay? So let's look at 30 and 40. So 30 and 40, they are here together once, and 30 and 40 are also here together. Okay, one. So they're also occurring 2 times. And then 35 and 25 is here once. 35 and 25 is here also 2 times. Then 40 and 25 is here once. 40 and 25 is here. So you can see each pair are occurring together twice, and they're all replicated equal number of times. And that's why I say this is balanced incomplete block design. So we can use our Agricola library package to do the balanced incomplete block design randomization process. This is the command that we're going to look at. And this command should then be able to give you the randomization that you're looking for. Okay, so the same command. So we create our treatment set, and then we bring this design.bib. This is balanced incomplete block design. You put your treatment set there, you put K, you put R, you put this. Each of these is actually defined. And then after running, you should be able to see. So here, for example, you can have that block 1. So block 1 here has 3 units. Here you have 40, 25, 30. Then you go to block 2, you have this, you go to block 3, and then you have block 4, and you'll have that. So R can help you to actually get for you the randomization and the field plan that you can use for that. Of course, sometimes it may not be possible for each pair of treatment to occur together equal number of times. As I indicated, it will lead to increase in the number of replication. You may have a lot of replication before that can happen. So we may not do balanced incomplete block design. We can go for what we call partially balanced incomplete block design. that will require less replication to construct. So instead of having all pairs of treatment occur 2 times in the same block, some pair of treatment may not even occur together. Some of them may occur 1, some of them may occur 2 or 3 or 4 times, depending on the importance of the comparison that you're trying to look at. So that is what we call a partial balanced incomplete block design. So different treatment pairs occur in the same block an equal number of times. And some pairs may never even occur together. So the mean comparison will have different levels of precision, because in this case, the number of replication will be different. So the different pairs will be, the different combination will come together or an equal number of times. And that means each treatment will occur together with the estimate for each treatment will have different replication and different precision, therefore. So this is an example of a partially balanced incomplete block design. So here you can see 1 and 4 occurring together twice, 2 and 5 occurring together twice, and then 6 and 3 also occurring together. But then 5 and 1 are actually not occurring together on this in the same plot, in the same block. 5 and 1s are occurring once. Sorry, I divided my thing there, so let me first clean it up. So 5 and 1s are occurring only once. There is no any other place again where 5 and 1 is occurring. And then 3 and 1 is also only once, and 5 and 3 also occurring only once. Okay, so different pairs are compared with different level of precision. So when you're designing, you need to ensure that there is a reasonable precision for all the pairwise comparison that you're likely to make. You can also do the design in R. And then I talked about the row column design from the 2 blocking criteria. So the Latin square is a complete block used to control variation in 2 different directions. So Latin square may actually be impractical, like I already indicated, because of the large number of treatment. So instead of having a complete square with equal number of rows and equal number of columns, you could actually have reduce the number of rows or reduce the number of columns depending on the circumstance that you're dealing with. So row column design, either rows or columns or both are incomplete. The famous row column design are what we call the Youden squares, where 2 or more rows are omitted from the Latin square design. In the past, it used to be very difficult actually to do randomization for row column design and for Latin square design. And most cases, what people would do, you go at the back of the textbook, and then you just pick one of the randomization process that is already done. But these days with the computer, it's a lot easier to do the randomization process. This is a nice example of a balanced row column design. So here is an example where you have cars. So here you have 7 cars. And then of course you're trying to test. So you have car number 1, car number 2, car number 3, car number 4. And then on these are the positions of the tires of the car. Of course, we assume that this is a car with 4 tires. So here position 1, position 2, position 3, position 4. I don't want to draw another picture at the risk of being laughed at. So what we have here is the different treatment that we actually have. So you can see if we look at position 1, of course, car 1 has all the cars, the 7 cars have position 1. So this is a complete, is complete. So here you also have a complete block. Here you have a complete block, and here you have a complete block. For you to have a Latin square, you need 7 by 7, okay? But in this case, naturally, there's no car with 7 positions of the tires, so you can only have a 7 by 4. So the rows, so the positions are complete, that all the cars will have the positions, but the cars only have 4 tires. So each car is actually, look at it, only has 4 positions within that. So this is a row column design. Each row is complete, but then each column is incomplete. So here we had to drop how many rows for complete? We dropped 3 rows for a completeness to have a full Latin square design. Then briefly, I talk about Yauden square, incomplete Latin square with 2 or more rows dropped or omitted. So finally, I'm going to talk about what we call resolvable or cyclic incomplete block design. So just going to take us a few minutes, and then I can convince my colleagues that we give you a break and then we meet tomorrow in the morning. So I could save you a few minutes as a bonus for being good students. Okay. So resolvable or cyclic incomplete block design. This designs became very famous in the day. I'm going to come to alpha lattice design. Okay, so these designs were became very famous in the 70s in the UK for simple reason that they were by law. The laws are restricting how many varieties you can actually have in your trial, and also restrict how many replicates you can have in your trial. So all the balance incomplete block design actually were not useful. So with the resolvable and design that we have blocks that are incomplete, in that the blocks have fewer units than the number of treatment that you have, but these blocks are always grouped in such a way that each group or block constitute one complete replication. So let's say, for example, we have 10 treatments. So I can have a block here, my block 1, that can only take 5 treatments. 1, 2, 3. Okay, so here I have my block number 1. Block number 1 can only take 5 treatments. Block number 2 can also take 5 treatments. Okay, so this is my block number 2. Each of them is an incomplete block. Now, what these guys were interested in, they still wanted to work with replication. So then these 2 blocks here now will constitute one replicate. So this is our replicate number 1. And this replicate number 1 actually sometimes is referred to as the super block. So blocks of blocks. So these are what we call our super blocks. Okay. Why was this so attractive? So this was attractive because field management was supposed to be completed, be done replicate by replicate. So people still wanted, they were still hanging on to the randomized complete block design where a block is complete and you have that. So instead of having, we have blocks that are smaller, but we combine, we have a number of them together to create a bigger block, which you call a super block, which is actually a number replicate. So it was actually attractive from the trial management perspective, in that you can actually weed a complete replicate today and then come and weed another replicate tomorrow. Field operation can also be conducted in stages. So I can do my planting block by block. I can do my weeding block by walk. I can do my data collection block by block. I can do my harvest block by block. So this one makes it really important. And then of course the other advantage would also that you can actually lose a complete, a complete block can actually be lost without actually having the whole experiment lost. Because once I have this, and then I have another set of replicate here, another set of replicate there, another set of replicate there. So even if I lose actually this set of replicate, I still have 3 more that I can actually still use. And then also realize that if we realize later on that these actually blocks are not very different, then we can actually drop the blocks and use the replicate as a complete block. And then we do our analysis with randomized complete block design, and you still be okay. You still enjoy all the advantage of RCBD, and especially those days where most of the calculation were done by hand or by simple calculators. Therefore, it was easier to actually calculate things like efficiency of RCBD compared to CRD than these days where everything is done in the computer without any problem. So that's why the resolvable designs were very popular in the 70s. So what we simply need in this case is we need to arrange our blocks, our smaller blocks, so that a number of them will actually constitute for us a complete replicate. So here, if you have a group of S blocks with K unit, K per block. So let me just go back to this example here. I can have this. So let's say I have here my block 1 with 5 units. Okay, and then I have another one here. 1, 2, 3, 4. And then let's say I have another one here. And then another one here. So I want to define what is K, what is S, and what is R. We only need to have that, okay? So here we have our T equals to 10. We have 10 treatments, okay? But our block is smaller. So the number of treatment, the number of experimental unit within a block K is equal to 5. So how many blocks do I need in order to get my 10 treatments? Can I see the answers in the chat? How many blocks do I need to get a complete set? 2, okay? So S here is equal to 2. Okay, so this is why we say now our T, which is the number of treatment, is actually equals to S times K. Okay, which is in this case is equal to 2 times 5. So K is what we call the size of the block, and then S is the number of blocks within the number of blocks that can constitute a replicate. So this is going to be our replicate number 1, and this is going to be our replicate number 2. This is going to be our replicate number 2. So the thing here is that we need to arrange our replicate in groups of S blocks with K unit per block. So the number of treatment is a multiple of that. S times K. And those who okay. So we then come with a special type of resolvable lattice or resolvable design that's called alpha lattice design. So they were part of the design that were developed by Patterson and Williams in 1976. And as I already indicated, the number of varieties and number replicated were fixed by statutory requirements. So if you're going to do an experiment, these are the number of varieties that you're supposed to try to test, and these are a number of replication that each one needs to have. So large number of varieties necessitated in complete block design and resolvable designs were required for proper management in the field. Okay, but there were existing resolvable design that did not always accommodate the number of varieties and block sizes required by trial. So these ones here were particularly then designed to take care of that. And the alpha lattice are actually the most common design used by plant breeders or people with high number of replications. So it still has the same thing, that the number of treatment is equal to the number of blocks times the size of the block. That's what you, so we always talk about S by K alpha lattice design, as that is what people always talk about. Okay, so except for the constraint that the number of treatment T is a multiple of the block size, there is no any other constraint to this. So you can always have as any types of alpha lattice design that you actually have. So you only need to define how many replicates you want, what is the size of the block, and then from the size of the block, you can tell how many blocks you need to actually have a complete replicates. So that is more or less what we have as your restriction. So alpha lattice designs have no limitation on block size. So the efficient alpha design exists for some combinations which conventional lattices do not exist. So it can accommodate an equal sample sizes as well. So this is the last slide, which is how to generate the Latin square alpha lattice design, which we are going to have a look at. I don't think we can look at it today, but I can spend a few more minutes to answer questions, and then we can run a small practical in the morning before somebody picks up. Okay, I am starting with the, what is the implication of P or partial balancing complete and in the use of minimum sample size in experiment, or does it apply to experimental studies? No. This one here, you cannot use this you cannot use this in calculation of sample sizes. What you can use in the calculation of sample sizes are going to be you use the idea of stratification. And when you're talking about the sample size, what I talked about in terms of the factors that you need to consider when calculating, when determining the number of replication equally applies to when you're calculating the sample size. So when you're calculating the sample size, the key things that are of interest are how much variability is there in the population that you're trying to study. What is like the error margin or that you want to, what kind of precisions that are you looking at? Also things like resources and also comes in there. Let me see the question. Okay. Difference between two-factor arrange in split plot arrangement in CRD and two-factor arrange. Okay, so you can okay. The whole idea here is let me try to see if I can answer this. Let me first end the show. Okay, so the idea of running a split plot design with running a factorial. So we can run factorial in a split plot. We can run factorial in a RCBD. So there are two different things here. So let's say, for example, we have let's just talk about, we have variety 1, variety 2, variety 3. I'm trying to use my thing that is trying to tease me a little bit. Okay, so let's say I have variety 1 and variety 2, and then I have say, what is happening here? I have variety 1 and variety 2. Let's say 1, 2, and 3. And then I have a spraying and spraying level 1 and spraying level 2. Okay, so in my if I look at the possible combination here, I can have variety 1 with spray level 1. I can have variety 1 with spray level 2. I can have variety 2 with spray level 1. I can have variety 2 with spray level 2. I can have variety 3 with spray level 1. And then I can have variety 3 with spray level 2. Okay, so these are my different combinations that I have. So I have 1, 2, 3, 4, 5, 6 combinations. So if I'm doing my randomized complete block design, what I will do, I will get my block. I'll get my block, and then I'm going to split my block here into 6 parts. 1, 2, 3, 4, 5. So here I have 1, 2, 3, 4, 5, 6. So I can randomly assign my treatment here. I call maybe T1, T3, T5, say T6, T2, and let's say T, which one is missing? 4? T4, okay? Where T1 is this combination. So here, I can repeat the same thing here. In another block, I do the randomization process. Now, so this is, I have my block number 1, block number 2, and then block number 3, and then I do the randomization process. So that's my factorial with RCBD when there's no split plot. I'm going to come and do a similar thing here, but this time I'm doing a split plot experiment. So the spray is going to be my main plot factor. So what I'm going to do, I'm going to have my block still. My block, 2 blocks. Okay. So what I'm going to do in this case is, the first thing is I'm going to look at these factors separately. The combination will come later on. So what I'm going to do, I'm first going to break to assign this. I'm going to divide my block into two big plots, what I call the main plot here. Then I can summarize here. I put, this is my S1 and that is my S2. Okay. And then I can also do the same thing here. This could be randomly, I assign these two. Okay. Now the splitting is going to come in. So let me get a different color. Okay, so I'm going to split this into 3. I split this into 3. I split this into 3. Okay, now if I look at S1, S1 here only has 1 replication. Okay, and S1 here, S2 here has only 1 replication. But when I come here, there is one S1 here where it is with V1, and then there's another S1 here with V2, and then there's another one with V3. So what you see in this case is that I have my one big plot of S1 and big plot of S2. Now I'm coming to put in my so I come in, then I put my, let me have a different color, sorry. So here I have my, let's say V2, I have my V1, I have my V3. Okay. Now the difference between here is that if I decide to say this is one replicate of S1, that's one replicate of a second replicate of S1, and third replicate of S1, I'm doing pseudo replication. So that's the big difference here between my assignment under split plot and my assignment under randomized complete block design. So you can see in this case is that the number of replication for the main plot factor differs between the 2 cases. Here you have fewer, the main plot factor is actually replicated fewer times. Here you have several times, the replication is more. So in terms of efficiency, in terms of precision, this is much better than this as far as S is concerned, because here S is replicated fewer times compared to in this particular case. I don't know whether I answered that question well. What will be the squared use denominator in calculating the value of p-plot? Now that one we can look at it differently. Any other question? What is the difference in the weather condition of the super block? The super blocks are like what you call a replicate. So if today the weather is bad, we assume that is going to affect everything in that super block. And that is the beauty of what we call uniform management or uniform condition. So it's actually important. That is part of the reason as to why they insist on having a replicate, because you can actually do your things on the replicate once and for all. Okay, so I think if there is no any other questions, then we can call it a day. What's the difference between alpha lattice and a Latin square? I think I've done that explanation. What's the implication of the weather condition for super block? I think I've done that. I'm working with 78 lines of rice. Is it okay to work with alpha lattice? How many blocks and how many replications recommended? Yes, yeah, with 78 lines, I think alpha lattice is probably the best. The number of blocks is, first of all, if you go to your field, you need to have a rough idea what kind of variability that you have. And this is also related to the questions that somebody asked, that if I have 200, I have 200 lines, how many replication? There are challenges with having replication when you have very many lines. So with 78, you could have 2 replication. That will still be fine. With the 200 plus, it may become very challenging. Sometimes people do what they call, use what called augmented design. So with augmented design, what you do is you you get what you call a local check, and then you can replicate the local checks rather than replicating there. So the other treatment will be unreplicated, but the local check will be replicated. So then you can use the variability in the local check as your background variation or experimental variation for testing. Yeah, so I cannot tell how many blocks you need to have, but you can determine if you want 2 replication, then you need to decide how many blocks, because depending on the size of the block. And remember, the number of treatment T should be equal to S times K. Okay, so in this case, for example, you can say 78, what is divisible by 78 that I can actually have? You're not recommended to read further that, okay. Yeah, I think that is. Okay, I think what I'm going to give you as assignment is to go and run all the scripts that you have there with you. Please download package Agricole, download and install, and then you run all my commands, the commands that I've given you, and then we can explain that tomorrow in the morning. Probably we need about 30 minutes in the morning to finish running all those things. And then the next activity will take off from there. So thank you for the day. I wish you a lovely remaining part of the day.